All right, hello folks. Welcome to the second day of PISCeS. I'm Matthew Yates. I'll be sharing the morning session here. My role here is mostly just to introduce the presenters. So I'd like to introduce Dr. Lauren Van Patter from the University of Guelph, who will be presenting on something that I think we probably don't think about enough as eDNA scientists.
Wonderful, thank you so much. Good morning, everyone. It's so nice to be here, and I really appreciate the invitation to come and share work on behalf of my co-authors, Dr. Elizabeth Clare and Dr. Quinn Rausch. We're sharing findings from a community survey, a research and eDNA user community survey on perspectives on ethical issues in eDNA research. I'm the Lang Professor in Community and Shelter Medicine at the Ontario Veterinary College, and my background is transdisciplinary human-animal studies, and so I look at cultural and spatial and ethical dimensions of human-animal relationships. So my point of entry into the collaboration was more around thinking about some of the ethics of multi-species research and how we understand other beings that we are studying. And so our manuscript is currently under review, and I'm just putting the information up here in case anyone is interested in looking this up later. And today I'm just going to present the findings from what we heard from the eDNA research community, as well as some more speculative discussion, and I welcome any questions or thoughts from folks at the end. And so just to set up a little bit of context for our point of entry into the study and why we were interested in conducting our survey, there's been growing awareness and dialogue around ethical complexities within research in a number of fields. And just as a couple of examples, we have sort of international protocols, best practices, guidelines that are increasingly being generated. A few of these are FAIR, so making sure that research is findable, accessible, interoperable, and reusable. And so especially when a lot of research is publicly funded, we want to ensure that public good is coming from these data. So understanding how data can be shared and made use of by others in the community as far as we can. And then there's additional guidelines, things like CARE, so the principles of collective benefit, authority to control, responsibility, and ethics, sort of stemming from an understanding of the collective rights to open data in the context of respecting the UN Declaration on the Rights of Indigenous Peoples and Indigenous Data Sovereignty. And so I think within these sorts of guidelines and others that are available within local contexts and as well as international dialogues and agreements, we see more consideration of a lot of the complexity when we think about animal ethics, when we think about environmental ethics and environmental justice, when we think about social justice and data democracy. There's just a great deal of complexity within these conversations. And within research, every step from applications for research funding to specifications from particular journals around best practices for publication can have different guidelines or expectations within this process. And so we were curious that a lot of the dialogue around environmental DNA collection is premised upon some of these benefits of the non-invasiveness of the method. And so when we think about animal ethics and some of the ways that we conceptualize ethical use of animals or other beings in research, such as the 3 Rs, the desire to refine or reduce or replace the use of animals in research, this idea of its non-invasiveness of the methodology, that we don't need direct sampling, whether that's lethal or non-lethal sampling that can have harms to individuals. If we're reducing these interactions and we're reducing the potential stress of these, then is this an ethically superior method? And so there's a reduced risk to the subjects as well as individuals undertaking data collection when there's less direct interaction depending on the taxa. But we were wondering, does this capture all of the complexity? Again, what about things like the potential heightened visibility of individuals that stems from being able to sort of capture presence or generate some sort of locational data, as well as some of these other less tangible potential harms around ecological ethics, environmental justice, social justice considerations. And so we were interested in what the research community's thoughts were around these questions. And as I'm sure all of you are much more familiar with than me when it comes to environmental eDNA. Some of the unique elements involve the expectation that data sets are placed within public repositories. Again, that this technology is increasingly becoming more and more accessible to a diverse range of actors. The fact that bycatch of non-target DNA is unavoidable within this method in a way that is different from many other technologies. And so, raising sort of the question around the complexities and way that the landscape of this technology is shifting, and asking what the data is being used for and by whom, and what are some of the ethical implications of this. So our aim was to explore perspectives on eDNA and ethics from amongst the research and end user communities, to determine levels of knowledge and concern for various... Oh, okay.
It sounds like the mic is dying.
The mic's dying? Okay, just one sec. Turn that one off.
Sorry about that.
No worries.
Please turn on.
Do you need battery swap?
No, the battery isn't here. Sorry about that.
Not at all. I'll take the chance to have a sip of water.
Well, they were both working a minute ago. I don't love that.
Of course.
Well, I'll change the battery on yours and we'll hope that that fixes it.
Sure, yeah. I think I need to.
Okay, let's give that a try.
Amazing.
Sorry
Thank you so
about
much. Yeah. And then we were also interested, or I'll present some of the results around if there's any sort of trends or associations between the type of work individuals are conducting or their demographics and their level of ethical concern for various topics. And so we designed a global cross-sectional mixed methods e-survey, and this was circulated through social media, targeted email lists, as well as research events where we felt we would reach the relevant members of the community. And so there are 23 questions, and these included a mixture of closed-ended and open-ended questions. The closed-ended questions were analyzed just using basic descriptive statistics, as well as my colleague Dr. Resch ran logistic regressions models to attempt to track. Okay. Track if there were any trends or associations, which I'll mention a few of when I share the results. And then the open-ended questions were analyzed qualitatively, just using content analysis to track the range of possible concerns that people are expressing on each particular topic, as well as a broader thematic analysis that was trying to trace patterns in folks' responses across questions. And so I'm going to share 5 themes around that. I'm just going to start before I dive into our results to share a few limitations of the study design and some considerations to bear in mind as I share the way that data were interpreted. So again, was largely done through opportunistic and purposive snowball sampling, so it's a non-representative sample of the research community. There's certainly overrepresentation of certain geographic regions, which I will share, and the survey was only circulated in English, and so of course that shaped the population of folks who were responding to the survey. So again, we can't take this as being representative, and was more of an exploratory study to try to capture some of the perspectives from members of the community, which could be used to design more random, targeting a more random sample of participants in the future of interest. And then some of the questions we dichotomized during analysis for the purpose of analysis, and also some folks wrote in answers to our multiple choice questions, meaning that we had not presented an exhaustive range of options. And so this just means there is some risk of over or underestimating demographics or associations that I present. We had 80 individuals respond to the survey, and within this, the majority of respondents self-identified as women, and between the ages of 18 and 40. And there was close to, an even split of those who did or did not hold graduate degrees. Their role in eDNA sampling was largely generators of eDNA data versus end users of the data. And the majority were also employed by universities with relatively fewer individuals who are coming from government agencies or private industry. And within their eDNA sampling practices, the majority were sampling within their country of residence. And I'll show another map of this in a moment that makes that a bit clearer. The majority were sampling within water. And target taxonomic groups were largely or primarily fishes, followed by mammals, and then relatively fewer sampling other vertebrates or plants and fungi. And so this map just shows that there are a couple of countries who had individuals responding to our survey who resided there, but no one reported sampling within those countries, and those are in light purple. Several countries had individuals reported sampling in the country, but nobody reported residing within those countries, and those are in green, where the majority in dark purple had participants to the study who both resided there, and there was sampling reported within the country. And also some geographic regions, Canada, the United States, the UK, and Australia were overrepresented in individuals who responded to the study. So in terms of levels of ethical concern that were reported by study respondents, in terms of the use of eDNA in research, most individuals responded that they were not concerned at all or only somewhat concerned with the ethical dimensions of eDNA use within research. And I think that given that the majority of respondents were coming from universities, it sort of makes sense that having less concern over the use in research contexts. There was relatively more concern of the use of eDNA within industry. And when we ran the logistic regression models, we found that individuals who conducted eDNA sampling outside of their country of residence had a 3 times higher odd of being moderately or very concerned with ethical issues around the use of eDNA in industry. In terms of the capture of non-target DNA, most participants were not concerned at all or only somewhat concerned with ethical issues surrounding the capture of non-target DNA. And again, within the models, older participants in this case had an 8.4 times higher odd of being moderately or very concerned with the capture of non-target DNA. And there's a few more findings. I'm going to weave through the qualitative results, but some of the key takeaways from the quantitative findings was that there was greater odds of significant concern amongst older participants, those who sampled outside of the country of residence, and those who used soil samples or identified primarily sampling in soil. Whereas there was relatively lower odds of significant ethical concern amongst individuals who collected vertebrate samples and those who reported preferring non-invasive techniques, which makes sense. And so when it comes to how individuals identified the range of concerns that they had across a number of topics, I'm going to start out with the capture of non-target DNA. And so there were 5 sort of categories or codes of responses that we found coming up in individuals' text responses to open-ended questions of why they were concerned or not concerned about the capture of non-target DNA. And so the first of these, I just have the codes here and then the number of times that this showed up across the surveys. And so one of the primary concerns was around this question of consent. And so primarily individuals were talking about the capture of human DNA. That's how they interpreted the question of non-target DNA when they were expressing written concerns. And so as one participant said, this may include human DNA. I would assume some people might not willingly provide that if they had the option. So there were a lot of folks saying, like, we can't consent to our DNA being in the environment and that being sampled, and that could be a problem. And relatedly, people expressed concerns around the potential privacy risks of this. So there's human DNA everywhere. It could always be lab contamination in the future. However, as assays get better, there could be privacy issues. And so again, we did find that a lot of these concerns around human bycatch were more speculative. Maybe it's not a concern now, but how could things change in the future? What concerns might that present down the road? And then related to this also, several individuals specifically noted this fear or concern around potential surveillance uses. And so as one respondent wrote, I would not want tools like airborne eDNA to be used maliciously, for instance, for government surveillance of different ethnicities. And so another respondent explicitly wrote like Big Brother type uses. So these, again, this was only several individuals, but did mention having these sorts of future speculative concerns. And then folks also wrote how the data could potentially be misused. So maybe this wasn't the intention amongst the researchers, but is there some risk, again, for it to be misused? For instance, I hope that technical developments in bioinformatics pipelines can remove potentially sensitive information before it's available for misuse. And so how individuals identified what could be potentially sensitive information. I think this included both human, but as I'll talk about later, potentially other taxa of concern for various reasons. But I do want to say that although there were these range of concerns listed, a lot of respondents to this survey did also say that they did not feel that this was a concern for them. So as one respondent wrote, eDNA of a person will only tell you if they were somewhere in the proximity of where the sample was taken. I can get the same information with my eyes when sampling. And so people were saying that they did not feel that there was a reason to be concerned about this. The second question we asked was about any concerns related to conservation. And most individuals noted that they were not concerned or only somewhat concerned. And usually this was because when they wrote in the answers was the non-invasive nature. So again, seeing this as inherently preferable. For those who did express some concern, this was largely for two reasons. The way that it could be used in species monitoring and the type of inferences made, as well as largely this fear of sharing the location of taxa of potential concern. And so in terms of its non-invasiveness, I like the non-invasive aspect of it. It makes it more ethical from that perspective, and so several individuals expressed this. There were some concerns around like what conclusions would be drawn from the data and how this could be used in species monitoring. For instance, basing conservation decisions on inaccurate information, for example, taking one sample and declaring an endangered species not present at the site. So there was some concern that this could be a risk of that. And then the concern that was expressed most often related to conservation were things like, I could see problematic uses of eDNA that could hinder conservation, such as using it to narrow down presence of species for illegal harvesting. So this was what we heard most often when it came to conservation concerns. We asked about concerns around data ownership, and this is where we saw more individuals were moderately or very concerned compared with the last couple of categories. And again, older participants had a 3.5 times higher odds of being moderately or very concerned about data ownership questions. And those who used soil as the main source of sampling were 4.4 times higher odds of being moderately or very concerned. And then there were primarily three sort of categories of responses within these, where folks both had this fear of public data misuse when data is made publicly available, but also showed a strong priority towards data democracy, which sort of led to this third theme of a lot of people acknowledging this complexity and challenges around how this could be negotiated in practice. And so in terms of this potential for misuse, again, once the data are published and publicly available, it can be misused through misinterpretations. Whereas on the other hand, quite a few individuals expressed, I think it's important this data is accessible and freely shared so the results can be interpreted as a collective. So again, the strong desire towards the democratization of eDNA data. And as a result of this, as one individual summed up, public posting of full reads could permit non-ethical analysis in various ways. However, public data sharing is a core ethical value. I see this as a legitimate conflict that will be difficult to solve well, so just acknowledging the complexity of the risks, but also the huge benefits of data sharing. We also asked whether individuals used any current guidelines within their work, and the majority of individuals indicated that they did not currently use any sort of formal guidelines or protocols. And the responses, I'm just going to share 5 out of the 7 sort of sub-codes here. But I will just flag back, even in the qualitative responses, the majority of individuals just commented about not having anything currently. And so there were some existing protocols, like trying to adhere to QFAIR principles for all eDNA projects, adhering to GDPR, so General Data Protection Regulations within citizen science projects where volunteer information is stored. And so people named certain individual regulations that they were following within their research. Several individuals noted that the only sort of ethics that they received were through their institutional animal ethics care or animal care committees or animal institutional ethics. And so this individual said even though it's not specifically involving animal manipulation, they still got approvals through the institution for the work. Some said that sampling permits, so we follow national and international research permitting and standards. These are at the country level, so they saw this as being something that would provide ethical oversight for the work that they were doing. A couple of individuals mentioned Indigenous-led guidelines, and so this one individual working in Ontario, New Zealand, talked about the Matra Era genetics guidelines as ways of implementing the fair and care. There were several individuals from Canada who mentioned using OCAP, and so the principle of ownership, control, access, and possession delineated by the First Nations Information Governance Centre. So these were more locally specific guidelines that were available. But the majority of folks said things like, not yet, but happy to consider good ideas. And then we also asked, do you have any solutions to any of the ethical concerns you may have expressed within the survey? And so it was a fairly even split of those who said, no, I don't have any solutions versus suggesting something. And so with those who did suggest something, there were a range of sort of more formal guidelines, industry standards, regulations or licenses, as well as several individuals, again, pointing to Indigenous-led solutions. And so, as one individual noted, they would appreciate seeing wider discussion in the field and code of ethics, and that would lead to explicit inclusion of ethical guidelines within regulations and permits. So this was what being envisioned. Someone else said standardizing methods of capture and protocols for data use. So again, more sort of industry standardization. Others said, more formal things. Regulatory frameworks, stricter laws governing usage will be essential to protect genetic privacy and limit use for biopiracy. And again, several respondents said things like science doesn't occur in a vacuum, so Tikanga, the Indigenous people's values and practices should be integrated within research. And then, so in addition to these, there were also a few emergent themes, and I'm just going to share about these really briefly. But several individuals in the written responses expressed the view that the benefits of the technology outweigh the cost, and we need to bear this in mind when we're having conversations about ethics and implementation. And so I see more benefit than harm in eDNA research. That sort of view was reflected a number of times. Others wrote that a lot of these ethical issues stem from the novelty of the technology and how rapidly it is developing. And so while the tools are in the development stage, I'd hate to see them used to make big decisions if they aren't optimized and as accurate as can be yet. And so several individuals said, we need to consider what sort of inferences are being drawn from the data and how it sort of intersects with perhaps what is perceived as more established methods. Others expressed this view that it wasn't really a risk within the data inherently. It was rather that sort of the quality of the methods that were being used or the analysis that were being performed. And so, for instance, if the assays or methodology were not accurate or rigorously vetted and erroneous conclusions are drawn, it could lead to inappropriate conservation strategies in this instance. And so it was more about how things were being mobilized than risks inherent to the data themselves, or to the methods themselves. And a lot of individuals, when asked again about broader ethical concerns, expressed this importance of accessibility and equity, sort of reflecting the preference for data democracy. Someone said, I'm concerned primarily with equitable availability of eDNA methods across political, geographical, and social boundaries. And so saying that there was sort of a perception of it reflecting, histories of use of technologies and conservation decisions being made by more elite sort of global north institutions and imposed on other communities. And that was a risk. And then again, this concern for an expression of need for more sort of formal guidelines. So ensuring robust ethical guidelines and strict laws will be essential to protect genetic privacy and maintain public trust. And now that I've shared a bit about what we heard from respondents, I'm just going to end by sharing a couple more speculative considerations along 4 topic areas that make eDNA research unique and things that my co-authors and myself thought are important to discuss amongst the community. And so again, I sort of started out by saying these benefits, and we think about animal ethics and things like the 3 Rs and the non-invasiveness, but we ask whether there are challenges when it comes to how data acquisition is being de-risked through the rapid development of the technology. And so, again, some of the challenges there are what are the implications when it comes to things like increased visibility, increased accessibility. Again, there are benefits here in terms of who can have access to understanding, ecological communities, but also are there concerns around what it could be used for, ranging from, again, as expressed by a number of respondents, potentially targeting particular taxa, highly trafficked taxa, or even things like, covert surveillance of agricultural diseases, which could have large economic implications. And so what does this de-risking potentially, what are the implications of this? The second is again around data ownership, which was one of the topics that individuals who responded to our survey expressed most concern around. And so the complexity when it comes to a data source where we cannot identify often the source of where the samples are coming from. And so how do we understand things like appropriately receiving permissions to sample when we can't trace the origins? So some respondents asked sort of these bigger picture questions of why should anyone own samples that belong to the earth? And when we think about, again, a lot of the research being publicly funded and having a sort of mandate that there's public good from this, and how does that sit in conflict with expectations around the economies that run on the principle of data ownership and patenting? And so how are these things negotiated? How can we think about the applications of things like the fair and care principles and equitable and mutually beneficial data sharing when again the localization of the samples and navigating those geographies within the communities, it can be a lot more challenging when we don't know the origins of samples. The third area was around public data disclosure. So again, a lot of respondents have flagged this requirement for public deposition, which has these benefits when it comes to democratization of data, but also there's a lot of questions remaining around how these data are protected, how they are, I'll get to that in a second. But when we think about the huge diversity in ways that different taxa are valued, the commodity value, the cultural value, how are these things negotiated in practice? And this raises questions for, is it appropriate to have universal guidelines or best practices that are applied, or should these be negotiated more on the local level? And then finally, we're saying around data security. So how do we think about how the way that eDNA data is secured, stored, backed up, archived? Are the risks of corruption of data, and within eDNA, this is potentially more complicated when there aren't always physical samples to go back to. And so based on what we heard from members of the community and some of that more speculative discussion, areas of future research could include, again, further surveys that were aiming for a more representative sample, available in multiple languages, using things like a sampling frame available, or incentives or things like that to try to reach a broader range of respondents. As well, the qualitative discussion is really coming from quite short answers within the survey, and so things like more in-depth interviews or focus groups can allow to have a greater depth of understanding of the complexity and nuance of people's perspectives, how these concerns sort of show up in practice, and what the implications are of this. And then as well, potentials for things like generating community consensus on priorities and guidelines. And so largely what we heard from respondents is there is support for further dialogue and generation of things like guidelines and best practices, and it can account for the unique complexities of eDNA research and use, and the rapidly evolving technological and social contexts that we find this all occurring within. And areas that I'd be interested in further thinking about and hearing perspectives on include how these conversations can be brought into dialogue with concerns that are emerging around environmental data justice, socially responsible conservation technologies, and Indigenous sovereignty and Indigenous-led solutions, and what that looks like in this context as well. So Beth and I would be really happy to take any questions folks have. Thank you.
Thank you so much for the talk. And it's something that I've been thinking about recently with some of our data. I have a very specific example that I would love to get feedback on how to handle it, because we do airborne DNA and we do plants, and we found marijuana in some of our samples. And so obviously in the middle of a park, or in the middle of a forest, someone has an illegal marijuana field or crop growing. And so with just publicly releasing that data, right, that now becomes something that if it got out, it could be used to kind of track. We have a GPS location, so it could be used to kind of find that.
Can we have that case study for the paper?
Sure. So I guess my question, I don't want to remove that from the data set, because now it's non-reproducible, so the number of ASVs goes down, the number of observed go down. Should I mask that from, I don't want to get people in trouble for something that's legal now in my state, but what do we do with that kind of data? And it's something that, we always laugh. We're like, oh, there's marijuana in our traps. But now with this talk, I'm like, oh, someone could get arrested because there's marijuana in our traps. And what are our responsibilities for?
So I'll make it more complicated, because this is exactly the problem. If that sample, as I understand it, is sequenced in Canada, you're fine.
Right.
If knowing that you sent it to the UK, they can't legally sequence it for you. Because marijuana is more tightly regulated in Britain than cocaine or fentanyl. So they cannot knowingly sequence it in a public lab in the UK, if you know that. There's a more complexity. We ran into that with a positive control, effectively. So yeah, there's no answer to that, but this is why we're doing it. Do you have an answer to that? You're the sociologist.
I mean, I would say, within my very different other community-engaged research, these are questions we take really seriously. What could be the implications on community members, and what are our responsibilities there? And I would think if there's the potential for harm, then if there is an ability to do something like masking that isn't going to have implications for the other conclusions you're drawing from the data, then I would say, from my very limited knowledge of this context, that we have to ask, say that is my responsibility as a researcher to make sure communities are not going to be harmed through the findings. And so it's the same with trail camera. I've used trail cameras in my research, and so like any capture of human data is immediately deleted from that, and I don't feel, again, that there would be any like ethical reason to ever like report anything happening with humans that I would find. But yeah, I don't know. Did you have a thought on
Yeah,
that
but the
topic?
parks do use trail cams to see usage and understand, human pressures. I feel like one of the things that we've seen over the decades of playing in new technology space is we always try to hold them to higher standards than our existing methods, which may or may not actually be appropriate. So to the marijuana example, I would say, well, the government already has high-resolution spot satellites that can read the brand of the cigarettes in the shirt pocket that you're wearing. So, if they want to spot your draws, they already do that with infrared cameras. It's not clear to me that this is really any different. And nobody in the UK would say, well, we can't look at a US satellite data because we might see a marijuana plant in Michigan.
It's true, and I agree with you totally on the higher standards problem. I had a reviewer write back about a paper, until you can explain every false negative, none of this data should be published. Find me any surveillance, biosurveillance system in the world that can explain every false negative. It's a weird higher standard, I totally agree. But I think the point of this is that some of these complexities are eDNA specific. So you only know there's marijuana in that sample because it was in your reference database. Had it not been, you wouldn't have asked the question, you would have put the data in a public repository, and the next person with marijuana in the reference discovers it for you. We have this concern about things like pangolins that I've had in my data sets. If you don't know it's there, you wouldn't scrub it, but someone else will find it. And that was raised a lot, was this idea, well, I may not know that I've disclosed the location of X, but the next person who sequences a more complex reference database discovers it. We don't even know what's in our noise. That was raised a lot. We're doing this without knowing what we're doing in a lot of cases, and someone else can find it. And that can be a good thing and a bad thing.
Hi. This is an absolutely essential talk, so thank you very much. I've done a lot of consulting work in written and GATT, and these concerns are raised a lot with local communities. Canada is not even a signatory to the Nagoya Protocols to protect the indigenous rights of genetic material. Collecting eDNA is collecting genetic material. Was there any feedback on that in your surveys?
I mean, I think, trying to think about folks' responses, it was more, a level of general concern around there not being guidelines that individuals felt were sufficient to direct their own ethical consideration of things like that. I don't think that was a specific example that came up amongst respondents in the survey. And I'm trying to think, again, there was global or representation from folks from a range of different contexts, and some mentioned specific, again, local protocols that they were following. But yeah, that's a really good point. Yeah.
I think we didn't hear a lot. Oddly enough, human bycatch was one of the least worry about things.
Yeah.
Partly because people who generate the data usually recognize you're capturing so little information in short reads that all you learn is humans. And I've never seen a sample that didn't have a string of human DNA in it. It's just a ubiquitous contaminant as far as I'm concerned. And the other problem is when people think about identifying a person, the amount of effort we would currently have to go through to do that, to somehow recover long enough stretches of chromosomes to get all 64 known needed microsatellite markers to do a fingerprint, your cell phone alerted you where someone was much faster than that by pinging a tower. So there's much better ways, so people aren't as concerned about that. The public is. My father-in-law asks me that question all the time. It's so technically complicated, I can think of a dozen better ways to find a person. Just walking around getting photographed continuously by every camera that's out there. So we're sort of comfortable with it in the contexts that we know. Look at the estimates of how many times you are photographed per day. But we're not as comfortable with it in the context where we don't understand how it works. And I think that's where the public is expressing more concern to me than the scientists do. Like the conversation with my father-in-law all the time. But that's my feeling on that, is how that kind of works. It's technically too complicated to be actionable now.
Thank you for a great presentation. This made me think of COVID wastewater monitoring and other monitoring for public health, which is much more directly related to human bycatch.
Yeah, I don't think anyone specifically mentioned that in this survey. Again, there was this like general sense of this fear of the potential of surveillance, but again, it was much more a future speculative concern of like, as you were saying, right now this isn't realistic, but this sense that, and it wasn't a great deal of participants, but some definitely said like, how could things change in the future where there could be a more, invasive sort of surveillance. Is that possible down the road, and do we need to think about that now with how the data is being collected and stored? But yeah, I think it was again much more of a speculative kind of.
I think we have a question for this one more. It's time with one. And the lucky one. On back a little bit to Nagoya compliance, we've done certificates of compliance for eDNA projects in a couple of countries now, and one of the big obstacles was the funder, say NASA, in the US, requires all data to be public and open access. And to get compliance, we had to promise that we would restrict some of like the site location, some of the sensitive information about endangered species as well. And so the loophole that we found was the local context annotations for when you publish sequence data. So it's not restricted, but it's like the wishes of the data providers are there. And I just wanted to put that on people's radar, that local context was helpful here. And even if there was a disagreement in how the data would be managed between the funder and the country where the samples are from, I'm happy to talk to people about the way that we made it work, but I am curious if you heard that the funder pressure for making data open was a concern for people as well.
From my memory, I think it was more when it came to like journal and publication rather than funder expectations, but I think I would have to go back to the data specifically mention that. But that does, I think, connect with both points. There were one or two individuals who, did say not less about human bycatch, but are there culturally, sensitive species that need to have a different level of protection at the local level because of specific community needs. So that's interesting to hear about that.
Yeah, and I think some of the more interesting concerns were the more subtle ones, like what if the data in public repositories like GenBank becomes corrupted? If you produce genomes, you have a source tissue you could go back to reconstruct that. We don't. We don't archive eDNA samples almost anywhere because nobody could possibly deal with the volume. Some people do, but the volume that most of us produce, you can't. So, like, if public data was attacked and corrupted, some of it is recoverable. And if you have a genome and you've taken it from a specimen in a museum, you can go back to it, that sample. But the type of data we produce is more vulnerable because we mostly don't have a sample to go back to. So there's an interesting, little more subtle concerns about this discipline specifically. That's what what we tried to do in the manuscript that's in review is say these are general sort of genetics issues or environmental issues. These are the eDNA issues, which are much more subtle, but very specific to what we do. It's interesting that humans didn't come up very much. It's the one everybody jumps on as the ethical concern. It was one of the least reported things by people who actually produce data.
All right. Thank you very much for a very interesting talk. Next up is Katherine Lunn from York University.
Good morning everyone. My name's Kat and I'm actually a PhD candidate in the Clare Lab at York University. And I'm just here to talk a bit about, my research on cavity nesting bee ecology and of course focusing on how environmental DNA can be used in order to do so. So, as I think we can all agree, pollinators are important. And a lot of previous research has really focused on the colonial honeybee or Apis mellifera, but obviously more native species are incredibly diverse and are also of crucial importance. Predominantly, previous studies on native species have been done so with lethal capture. So these use like pan traps or collection of larvae from bee homes, but they're killed in order to study them. However, the increase in knowledge of environmental DNA, it really gives us more options in order to study this ecology without it being so invasive. So, in terms of eDNA and its use in ecology, in wild bee ecology, there are many different sources of environmental DNA. So one of the really well-known ones is collection of pollen from on the actual bee species themselves. We can also collect insect DNA from the different floral resources that they use. And then more relevant to my research is the use of bee homes or trap nests and the use of the biological material in there. So previously, using bee homes as a source of environmental DNA has mostly done studies on the floral resources that they're using. However, there have been a few studies on the other taxa that you can find in there, including Kuhneman et al. In 2023 did a study on the different bacteria in there, and then actually my own research for my master's, which focused on the fungi that we can find in there. So this is a way of looking at the bee ecology using the biological material. However, it hasn't actually been shown that we are picking up exactly the same things from the larva and the biological material itself. Yes, it's definitely a source of this plant material, but can we pick up the same thing between the larva and the bee material? So, biological material in homes. Character nesting bee homes, so this is an artificial nest, and this is what a brood tube looks like. And essentially, they find a little plant stem or a hole, and they use it to raise their young. So, unlike honeybees, females work alone and build their own nest. They gather pollen and resin, and they take it into the home, and they leave a little brood, like food ball there. They then lay an egg next to it, and they seal off a little chamber. They then do this continuously down the brood tube. So the mother lays all her young down the bee tube, and the bee nests are built with different materials. This can be mud or leaf pieces, plant resin. It just depends on the species. So the young then overwinter not always, but overwinter as either larva or as a pupae, and they emerge normally the next season. Sometimes they can do it later than that. So within this brood tube, we have a larva, some pollen, feces from if the larva's consumed the pollen, and then all the material there. Also, it'll have all the bacteria that live there or parasites that live within that home. So there's loads of DNA, biological material in here. So like I said, what I wanted to do was see whether we can use the biological material that's found here rather than larvae themselves and pick up the same things. This would be like a way of doing this without killing the larva, be able to use the material, and we'll pick up the same ecology. We're going to pick up the same fungi, plant, and bacteria. Can we do this with the biological material? So this is my initial question for my thesis. In order to do so, 100 of these artificial bee homes, each with 30 brood tubes, were placed around Guelph and Cambridge over the flight season of 2022. They were put out in May and collected in September. Once they were collected, so this is a brood tube, they were dissected, and we took out the larva and then the associated cell contents. So we then had this paired sample design. They were, so we had 1,483 paired samples in total. These were sent away, and they were DNA was extracted from both samples. They were amplified for the four different target regions. So our focuses were finding out what the larvae were, so we use CO1 for insects, And then for what they were interacting with, we did 2 different types of ITS for plants and fungi, and then 16S for bacteria. So, what we sort of found, so in order to identify who the occupant was, we used the highest read count above 1,000 reads, just to make sure that we were picking up definitely what was the occupant. And then in order to make sure that this was really giving us the occupant and not just random, like just some DNA that's found its way in there, what we did was we considered what the dominant read count was, what was the highest read count, and then if there was a secondary signal, because there often wasn't, what was the read count in that? So in the larval samples, well, in both types of samples, the dominant read counts is in the tens of thousands. So the plus or minus is like the range, so it's pretty much always in the tens of thousands. If there was a secondary signal, it's in the hundreds. It's way below the dominant read count. So we felt that we could really be sure that what we were picking up was the occupant of this brood tube. So in total, we managed to identify 2,190 samples of the worth of occupants. Of these, 1,158 belong to the Megacalidae or Peletidae families, so these are bee families. And then the other families that we picked up were like wasps or moths, or like other insects, arthropods that use these homes for themselves. So 990 of the bee samples were of a pair, so we could compare them, and then we ended up aggregating them to the brood tubes, to the brood tube level. So I had multiple cells per brood tube. We ended up aggregating them into each brood tube, and this was just because we can't really guarantee independence when it's a larva, it's an offspring of a single female. So what we found. So this is looking at the proportion of genera that was found shared within a cell. So it's a direct comparison between the larva and the cell samples. So if we found that the genus was in both the larva and the cell sample, then it goes into shared. If it wasn't, it goes into cell or larva. So for fungi and insects, we can kind of see this clear majority that we can see the genus was found in both sample types. However, in microbes and plants, it does look like a lot less. But this is a direct comparison of every single genus. It's not the amount of detections. So let me just explain, I'll explain that a little more. So I'm going to use plant as my example today. So for here, it looks like only 40% roughly of the plant genera were actually found in both sample types. However, when we consider the top, like most common genera that we found of plants, so these are the top 10 most detected genera. So in cell, these genera make up over 50% of the total detections. Like over 50% of all the detections we made for plants were of these 10 genera. And what's nice is that we can see similar distributions of each genus in both cell and larval samples. When we do a little statistical comparison, so I used McNemar's test to see if plant DNA was equally likely. So for each genus, how likely are we to find this genus in the cell material as we are the larval material? So the highest difference that we found was in prunus, and that's like 17% difference, and none of these are significant. So we're not finding a significant difference in the likelihood of detection between our sample types. So this is great, picking up the same things. When we consider the community level, looking at the variation within each within like the plant genera. So we see around 28% variation, which is really low. And really what you're looking at in this graph is the overlapping ellipses. We're seeing the similar things in both communities. We've got high overlap here. And then to test it statistically, I used a permanova, and we were looking at is detected taxa explained by sample type? but also because we thought these might have an impact. Is it explained by occupant species, the habitat it was, or the bee home itself? And what we found was none of these things, none of the other things impacted it at all. There was no significance here. However, sample type was significant, but it explained less than 1% of the variation between the sample types. So though it's significant, it's actually explaining very, very little of this variation we're seeing in the communities. And we're seeing a similar thing across the fungi and microbial communities. In the microbial PCOA, you can see this like high overlap that has a similar spread to the plants. In the fungi, there is clear clustering, but whatever the clustering is, it is not done by sample type. We can see both samples of both types in both those clusters. So we are seeing some formation here, but whatever it is, it's not by the sample type. So, takeaways from this research. We could identify the same occupant in 85% of cases. We could reliably recover like the core plant communities that were of these bee species. And the same is true for most of the most common genera in fungal and microbial communities. So we're really picking up this similar things. And it really means that we can use these we can use this material in order to study the ecology of these bee species without using the larvae themselves, without disrupting their life cycle, and they can go off and live after they've been sampled. It means that we can do large-scale ecological analysis without lethal sampling. Yeah, and that's everything I have to say. I just thank you to all the people that helped me, and thank you for listening.
I think we have time for one question, if there's any from the audience.
Yeah, so guessing you're studying here solitary bees, which are like their lifestyle and cycles are quite different from honeybees.
Yes.
So what's is it not possible to take those pollen pellets to do the study? I'm guessing that, does the cell for these solitary bees get sealed right after?
Yeah, they get sealed immediately. So previous studies have used the pollen ball, and you can look at the floral resources from that pollen ball, absolutely, and then you can see the bacterial and fungi that are within that. However, if you do that, you're actually taking away the food source of the larva, so that's another problem. Whereas if we can use this biological material, we can take a little subsection of it and get the same results, and that's really the positive of this.
All right, our next speaker is Nina Garrett, also from the University.
Okay, yes, so my name is Nina, and I'm also a PhD candidate in the Clare Lab at York University. I don't study bees. What I look at is using airborne eDNA to study bat roosting ecology, which is a little bit of what I'm going to be talking about today. So in order to talk a little bit more about this, I have to go back to my master's. And what I did for my master's was I looked at whether or not we could use airborne eDNA to actually detect what bats were in certain roosts, kind of as a proof of concept thing. It ended up working really well. We deployed our samplers in a variety of cave types, including trees and caves. And we detected almost all of the known species in these caves. The study site we were using has been surveyed for almost 2 decades, I think. So we have a really good species list of what should be in these caves, and what we were detecting was matching that. There were some exciting exceptions, most notably the white-winged vampire bat, which based on range maps and habitat has always thought to be in the area, but has never actually been physically caught. So hopefully in the next few years of surveying, they'll physically catch that to confirm that detection. But what I noticed in some of the specifically cave roosts when I was doing my master's work was a suggestive pattern of differences in detections between the day and the night. And so this led to this sort of overarching question that I wanted to explore a bit more in my PhD, which was, can airborne eDNA detect changes in community composition between the day and night in neotropical bat roosts? And to do that, I went back to the same study site, which was in the Orange Walk district of Belize, and I deployed my samplers in 5 caves, 2 excavated tunnels, and 3 natural caves, from the last week of April to the first week of May 2023. So these are some of my roosting, well, these are my roosting sites. We have the excavated tunnels, which are actually into like looters' tunnels into Mayan ruins that the bats have started to use to roost, and then some of our natural caves there. And what I did for these was I deployed either 4 or 6 samplers in the roost, depending on roost size, but also access. So there were a couple that I physically couldn't get into because the entrance was too small, so I had to reach around and place as many as I could reach, and I am not a very long person, so that was not very many. And I ran each of these samplers for 3 days and 3 nights, so a total of 24 or 36 samples per roost, depending on the number of samples. I then took these filters back with me to Canada, and a very quick overview of what the lab work looks like. We take that filter disc and we extract it using an eDNA extraction kit from QIAGEN. I then amplified it using a General 16S mammal primer, sequenced it on Illumina MiSeq, and ran it through the Data2 pipeline to get ASVs, which we then run through BLAST and use the NCBI database to match to species. I then used that presence-absence data to generate a series of occupancy models. I'm going to briefly go over the various levels of these. The first level is occupancy. This is the probability that a species DNA occurs at that site. The second is the sample level. It's known as the availability probability, and it's the conditional probability that your target DNA occurs in your sample, given that it was present at the site. And then the final level is your PCR replicate level, or detection probability, and it's that conditional probability that you actually amplify the DNA, given that it is both in your sample and at the site. So I ran these hierarchical occupancy models for our 7 topmost detected species. We have Desmodus rotundus, Natalis mexicanus, Trachops, that, Glossophaga mutiga, a molossus species, Corolla crispus, and Psychotrix luminata. You'll notice that the molossus is at the species level, so we keep our molossus species detections always at the genus level. They are particularly difficult to differentiate using mitochondrial DNA, sometimes only differing by a single base pair. And so when you use an amplicon length of like 90 base pairs, that can get a little dicey. So to be more conservative, we always keep those to our genus level, even if we do have 100% species match. So the first type of models that I ran were null models, so no covariates included. For almost all of our species, we had a high occupancy probability, with the exception of Psychotrix. Not entirely surprising, to be honest. While these all are quite common species for the area, Psychotrix isn't necessarily known to be a cave rooster. We only detected it in 1 cave, and I do know it was in there because I saw it in there every time I went up to survey, but it makes sense that we're not detecting it in all of our caves, and that will drive the occupancy probability down. But the key point for this is we can detect most of the common species if they're in the caves. For the next level, we had a relatively low availability probability. So DNA actually available to be caught on a sample was relatively low. And so what this kind of says to us is it's important to have multiple samplers. If you miss it on one sampler, the more you have, the more likely you are to catch it on another. It should be noted, though, that this particular level does also intersect with a lot of behavior. So behavior is going to impact shedding rate and things like that, which is going to impact the availability of DNA to be picked up. So our final level, the detection probability, again, goes back up. In fact, in a lot of cases, it's higher than the availability probability. So what we took this to mean was even if it's there's a low chance of you getting it in your sample. Once you get it in your sample, you're getting good amplification, and there's a good chance you'll amplify it in your PCRs. And so now to actually start to answer that question that I proposed at the beginning, I ran another type of model with sampling time as a covariate. So this covariate was, did we sample during the day, did we sample during the night? And this was included as a sample level covariate, so it would be affecting that availability probability. And for most species, it did have some sort of impact. And this impact did match some of the observed behavior we know and have seen for the species that are occupying these caves. So for those who had a higher probability during the day, if you look at Carolia and Psychopteryx, these two are most detected in those excavated tunnel roosts, which are in and out. When we go in there, the bats are in there, they're flying around. That is their day roost. So they're in there during the day. It makes sense, we're going to have a higher chance of detecting them then. They are likely then leaving the cave to go forage at night and no longer in there. For others, it was the reverse. So the availability probability was higher during the night. This, again, matches observed behavior. The best example of that is for our Nautilus mexicanus. They primarily roost in what we call the schoolhouse caves. And these are kind of what we expect to be larger openings to a more, a smaller network of crevices under this big hill. So when I go in to sample these caves, I often will only see one or two bats, or won't see them, but will hear them, and it kind of goes back into this crevice that I can't get through. And what we've observed them doing is when they're going out to forage at night, they will swarm in that part of the cave where we can actually put our samplers before they leave. So again, it makes more sense that you're going to detect them at night when they're moving about, shedding their DNA. And so a little bit about take-home messages. Multiple samplers are beneficial, but even if the eDNA that you actually have available to pick up in a sample, detecting a species at the PCR level can still be quite high, so you're getting that good amplification. The second take-home message, sampling time does matter. It is impacted by bat behavior, so you do have to take that into account. But it could also suggest community composition changes happening at a very short time frame. So we know that bats are very mobile, and they will change roosts quite often, sometimes over the course of the day, so they'll roost at night somewhere else than they do during the day. And some of the behavior patterns that we see that are reflected in our data could be indicative of that. And then for next steps. So the biggest thing that I want to try next is get more sites. I'm sure you saw some very large error bars there. I only had access to 5 caves, but I have another field site where I have more, which will hopefully not only make those error bars smaller, but make our results a little bit more reliable. And I'd also like to try either a multi-species model or including presence of other species as a covariate, because for a lot of bats, whether or not a certain species is also co-roosting there can be a factor in roost choice. So thank you for listening, and I'm happy to put questions.
Thank you for the talk, Nina. Your master's work was a big inspiration for my own air sampling equipment. And I had a question, if your setup, did you ever experience rodents or other animals destroying your filters of the caves? Because I've had problems with wood rat taking the filters for their bedding.
All the time.
Okay. And
so my other field site is in Northern California, and half the time I come back down there and the filter is like across the cave. I've lost them, they're gone. Can't find them. Another time they chewed through the wires of my equipment, and then I don't know where they took the wires, but they were gone. So it was just my little sampler with like this much wire left and then my battery with the plug in it. All the time, yes.
Yeah, it looked like the talks yesterday had it figured out. Their filters looked a little more protected than ours.
Yeah, I need to figure out how to put it on a stick. I think that will help.
A very tall stick.
Yeah.
Thank you.
Hello. I was wondering if you put also sound recorders to associate the activity of the bats with the eDNA, no?
So not for this study. I am for my work in California. The thing about a lot of neotropical bats is they don't make species-specific echolocation calls. Some of them do, but so in terms of, I think a lot of people say you should use it as like a confirmation of species presence, but I think you're getting at for a measure of bat activity. Is that correct?
Yes.
Yeah, so that is something that I'll be doing in California. I didn't do it for this, but it'll be one of my potential covariates for my California work.
Yeah, because they do have, we just published, neotropical bats. And the second question was if you put also your samplers outside the roost, to be able, I want to see if with those samplers you can really detect outside in the woodlands, the bat activity or presence.
So for this year, yeah, it was this year. The one cave we put out in an attempt to create some sort of field native. We put out just one of our little, it wasn't battery powered, it was just a little sampler with a filter. So basically kind of like an environmental sample. And we did actually detect a tree roosting species that had not been caught there before. We detected it it must have been roosting in the tree above. And then we thought it was contamination, so we got rid of it. And then the next year, we caught that back. So yes, we have done that. We didn't realize we were actually catching real detections, but we have done that.
Thank you so much, Nina.
Our next speaker is Katherine Dubuc from the INRS.
Hello everyone, thank you for being here. My name is Katherine and I'm a master's student at INRS in Quebec. And today I will be presenting my research, Municipalities Monitor Aquatic Invasive Species and Support Recreational Tourism using eDNA. So eDNA methods are well known for complementing traditional biodiversity surveys. Here, the research world believes eDNA is a powerful tool, and we're even working together towards standardization. But before organizations and municipalities can adopt a standardized method, they first need to see eDNA as a viable option. Right now, when municipalities want to survey their biodiversity, this is what they see. Many options are offered to them. Some have never heard about eDNA or don't quite know how it works, while some simply don't trust it yet. That is the gap my study is trying to address. We think eDNA should be highlighted to municipalities as a way to simplify and extend their biodiversity surveys, then assess biodiversity across all of their possible ecosystems, and finally support the implementation of conservation policies eventually. This leads to my objective, which is to use eDNA in 2 small municipalities to respond to their specific needs. So on one, both municipalities want to assess the biodiversity in general on their territory to educate and raise public awareness, but they also have their own specific needs. On one side, Rouyn-Noranda wants to monitor the spread of the spiny water flea across different lakes through early detection. The spiny water flea is what you see there. It's a small crustacean that is invasive. It came from Eurasia a few decades ago, and now it's established in the Great Lakes region, and now it's starting to establish at the west of the Quebec province, exactly where Rouyn-Noranda is. And it is problematic because this species eats all of the zooplankton in the lakes, which is food for the juveniles of fishes like the trouts and the salmon. So it's problematic for the ecosystems, but also for the fishing industries. On the other side, PISCeS Sandre wants to monitor past stocking efforts in their ZECs. A ZEC is in Quebec like a controlled wildlife management zone where fishing is permitted. And they want to do that to improve the management of recreational activities. So I'm going to start with Rouyn-Noranda. This is my sampling plan in the city. So I sampled water in lakes of biodiversity reserves, recreational lakes, and in the municipal water intake that you see in blue. And this lake was very large, so I took 2 samples at 2 different places to try to represent the most as possible. And in every station, I sampled 3 times 1 liter of water. Then for the methods, I filtered my water and I did my eDNA extractions. And then I proceeded to do different analyses. So I did non-targeted metabarcoding analysis, targeting the CO1 region, targeting metazoa, and the 12S rDNA region, targeting mostly fishes. Then I did my sequencing with Illumina and my bioinformatics with the BARC pipeline. In parallel, I did some targeted qPCR analysis on the targeted species, which was the spiny water flea. Let's move on to the results. This graph shows my results for my metabarcoding with the CO1 primers. So for every site, we see the number of species I detected and also their different phylums. So we see there were annelids, arthropods, chordata, etc. So we see those primers succeeded to detect great biodiversity in a few sites, but we see there is a huge variation depending on the site. And the best example is on the far right, Lac Dubuc is a municipal water intake. Those 2 samples were in the same lake, you remember? So we can see there's a huge difference in the results depending on where the sample was taken. This other graph shows my result for the 12S primers. So here no abundance is shown. It's only the different species I detected. And those primers detected a total of around 20 fish species distributed across the lakes. So it worked pretty well. Now for my qPCR, I had no detection of the spiny water flea in any of the analyzed lakes. But as I cannot confirm the absence of the species only with this result, I will be optimizing my sampling in 2026 for a better representativity using 2 methods I will explain at the end. And I will also resample some of these lakes, but I will sample other lakes of the same watershed too. Now let's move on to PISCeS Sandre, who wanted to monitor past stocking efforts and assess the biodiversity. So here's my sampling plan. To do that, I sampled in 14 sites in the municipality, as well as in an outfitter and in the ZEC in blue. So amongst these, there are small rivers, larger rivers, lakes, and there's also a marsh. And there are sites in the forest as well as in the urban area, so many different ecosystems. For the methods, I really follow the same methods as in the other municipalities, so non-targeted metabarcoding with the CO1 and the 12S. But I did my qPCR analysis on 5 different species this time. So the brook trout, the Arctic char, the Atlantic salmon, American eel, and the moose. And those are species chosen by the municipality. For my results, this graph is the same graph as earlier to show the metabarcoding with the CO1 primers. We see the variation is even bigger this time, especially because my river sites have a lot of species detected, almost 100 for certain sites, and the marsh has only a few species detected in it. Now for my 12S, the same graph as earlier, we see again that there are more species detected in the rivers, and that was already said in the literature by Daner and collaborators especially. They said that rivers tend to contain more DNA because they represent a larger zone or larger area than the lakes that are more local. Now for my qPCR results, this is for the brook trout. Here this graph shows the log of the concentration of eDNA for the species in every site. So for the brook trout, we see it was pretty much everywhere as expected, except in the marsh, of course. This is, I'm sorry, it's very small, but this is my result for the other species. Well, it corresponds to what we were expecting. So we expected detections for every species in those sites. So far with what I have done, I think eDNA can easily be used by municipalities to monitor invasive species, but also species of recreational tourism interest. But the active sampling like I use tends to give an uneven representation of the species richness. As we saw in the rivers, a lot of DNA was detected, and in the lakes a bit less, and in the marsh barely any. That is why next year I will sample more water bodies, but I will also try new techniques. So the composite sampling, it consists in taking as many small amount of water all across the water body and combining them in a single sample with a single filter representing the whole water body. And passive sampling in the water, so in that case I will put a filter inside a pickleball and I will attach the pickleball inside the lake and leave it for at least 24 hours before taking back, so this will have a better temporal representativity. So on this I'd like to thank my lab, my partners and my contributors. Thank you.
We do have a few minutes for questions, if there's any from the audience.
Hi, thank you. You mentioned at the beginning that you wanted to really engage more with municipalities, and I'm wondering, have you shown them this data, and what has been their response? Have they been, excited about it? Has it interested them?
Yes, I already showed my data to the municipalities and they were really excited about it. They had a lot of questions, of course, and the goal was to show them, like they gave me their ideas and I did only what they wanted me to do. And then I showed this for 2025 and they decide with this what they're gonna do for next year. That is why we use meta-barcoding a lot in 2025, because they wanted to have an overview of what was in the territory. And 2026 will be more qPCRs to really confirm some questions that they had, like why did we detect this species there? So it's really more precise. But yeah, I think it helped them overall.
So you think it kind of improved their trust in eDNA overall?
Yeah, maybe not meta-barcoding, but in qPCR, yes, and I think they liked it.
Most eDNA studies talk about how this is better to complement instead of replace other methods. You're making an argument here that this is the one technique that municipalities should use. And there are some studies looking at comparative effectiveness for detecting spiny water fleas. Do you think municipalities really should choose this tool and just this tool?
No, sorry, this is not what I wanted to say. I think yes, complementing both is better, but for smaller municipalities, they don't necessarily have the resources to do all of these surveys. This is why I'm saying that eDNA could help them get an overview at first, or help them do surveys that they cannot have the resources for. And in Huaynahanda, the ministry of the province do surveys of the spiny water flea, but they use eDNA and they also use nets. And our goal here was to do this survey with eDNA in lakes that the ministry don't do, to extend the surveys. Yeah, but I'm not saying to use only this.
We have time for one more question, if we have anyone. All right, well, please join me and thank you.
Hello everyone, I'm Fidji Sandre. I'm a postdoctoral researcher at INRS in Dubuc, and I'm working in the Valley Lumber Research Group. Today I'd like to present a project we've been conducting in collaboration with a Keynote community of Dubuc. Our main objective is to monitor biodiversity in Arctic region in northern Quebec. So Arctic ecosystems are unique. They are shaped by extreme environmental conditions. And the organisms that live there have developed remarkable adaptation strategies in order to survive this condition. However, this fragile balance have been threatened by climate change. As you may know, Arctic and subarctic regions are warming faster than the global average due to several reinforcing processes. It's called Arctic amplification. So the hydrological cycles are shifting, and the organisms that live on them are shifting as well. And the local communities are the most directly affected by this change, as they depend on this organism for their cultural practice and food security. So we need to establish robust conservation strategies in order to preserve biodiversity, and for that we need a strong assessment of this biodiversity. So as you may know, traditional methods such as that are based on observation of or capture survey are limited, especially in areas that are so vast and difficult to access as Arctic environments. So that's why we choose to use a eDNA-based tool to characterize this biodiversity and provide extensive data collection. So as I mentioned earlier, we are developing this project along with the community of Dubuc, and we have two main objectives. The first one is to assess aquatic diversity in a section of dynamics you can see in the yellow area. And the second objective is to monitor the presence of a few emblematic species that have been selected by the community. So we start with two helicopter-based sampling campaigns in the territory. We collect water from 45 sites in 3 days along the George River watershed and the Brook River watershed here. We've been conducting this sampling campaign along with youth from the community when we train for sampling practice. Then we extract DNA, and we also check for the presence of PCR inhibitors. We've been following Canadian Standard Association guideline 214 and 219 that we have developed in previous projects. And then we use both non-targeted analysis with metabarcoding and targeted analysis with QPCR. So for the metabarcoding analysis, we target 12S region of mitochondrial gene using MyFish primer. And then we prepare a sample, prepare a library for sequencing, and sequence a sample with MySeq DNA. And then we use a large bioinformatic pipeline for data treatment and taxonomic assignment. So here are some results for the metabarcoding analysis. All the dots you can see are correspond to sampling size, and the darker the color, the more species we've detected. We have detected 26 unique freshwater fish species in the watershed, and 35 if we include the marine site.
we can see here two areas of greater biodiversity. One is the river mouth and one is the south. For the one in the south, it could be explained by more easy environmental condition and greater lake system. And for the north greater diversity zone, it could be explained by the downstream transport of eDNA that has been produced along the watershed, and also because of the confluence with marine sites. And we detect DNA from marine species also at this site. We also detect some species that are being classified as vulnerable in Quebec or endangered in Canada, such as Arctic sisal, free-spine stickleback, and also moon snakes. However, meta-reckoning analysis do not guarantee the presence of this organism. So the next step will be to try to detect them with target analysis to be sure they're here. Speaking of target analysis, for each organisms, we develop a species-specific primary probe by researching a unique region in mitochondrial genome of individual species and comparing it to all symbiotic species. For the design of primary probes, we have several validation steps. We start with synthetic DNA, Dubuc. Then we use animal tissue, some of which have been provided by the community. And then we have field validation steps. We also follow CSA guidelines that I mentioned earlier. And then we check for the presence of the DNA for target species like PISCeS with health technical replicants. We also use this the same procedure in collaboration with Katherine Lunn to develop more than 135 detection kits for North American species. So for this project, we developed 12 kits for emblematic species that have been chosen by the community. Some kits are still in development for beluga, seal, and muskrat, but they doesn't use a result from the other species. Starting with mammals. So we did not detect black bear and beaver otter. However, we did detect Canadian beaver at several stations. You can see few stations in the south that are matching the distribution range established by the Quebec government. And we also see another station in the north, but we did not see any beaver around this area. But this station corresponds to the location of the village of King Dubuc, and it's not unlikely to see some traces of beavers there. And the other hypothesis could be the downstream transport of the DNA produced along the watershed and could be accumulated in this area. We also see some caribou DNA. So for protection reasons, these are not the real location of the caribou as requested by the community. But those locations correspond to the detection of correspond to GPS location of caribou that are followed by Quebec government using a GPS log. We also see two other detection stations in the north, and the same hypothesis as for beaver could be formulated. It could be downstream transport of DNA. And then for the fish species, as we did non-target analysis on the species, I have the result for both. So non-targeted analysis results are shown in red, and targeted results are shown in white. So Atlantic salmon, we've been detected it in a lot of sites across the watershed. And we have been able to confirm the non-targeted analysis as a metabolic detection with targeted analysis, and we also detected in more stations with targeted analysis. The same patterns have been observed for lake whitefish and also for white sugar, with more detection with QPCR. For Mozambique, we only detected in 3 sites, and it was the same for QPCR and Dubuc. However, for hard teacher, the opposite pattern have been seen with more detection using metallurgy. However, QPCR assays are designed to amplify only the target species DNA. Opposition with metallurgy that could have some bias and amplify DNA from closely related species. That could be the case here because Sabinus is closely genetically related to Sabinus fontivenis, and they are not too difficult to differentiate. So distinguish from the Clavex marker. So I will give to I tend to give more weight to the QPCR result process. So in conclusion, we've seen here that DNA could be a really useful tool to characterize biodiversity in Arctic regions that are difficult to access. And it's also a good example of collaboration between scientists and local community. We detect more than 35 unique species and also vulnerable species within metallurgy. We've been able to detect caribou, beaver, and a lot of fish species using QPCR, and even shows that QPCR could help from metallurgy for some species, showing that the two methods are complementary. From some perspective of this work, we have to confirm the metallurgy signal we have for some Sabinus species by doing QPCR analysis for Sabinus nomenclature, Sabinus fontinalis, and we also have to develop QPCR assay to confirm the presence of vulnerable species that we may have seen using metallurgy and maybe contribute to the protection. Okay, well, it was supposed to be the acknowledgements, so thank you very much.
Hi, that was great talk, thanks. A couple of months ago, there was discussion about setting up an autonomous eDNA lab in Dubuc at the Nuremberg Research Center. Is that something you're involved with? That could, it would be nice to have your expertise involved.
I was not aware of. I know there is another project in the lab that was connected with the community. I'm not involved in this. Not yet. I'm supposed to.
Thank you. I'm really curious, in your reference database for the metabarcoding, did you include some sequences for some of the mammals that you were interested in targeting? Because I know from my own work that sometimes primers that are putatively designed to target fish can often pick up secondary detections of aquatic mammals, so that could be another way to validate the qPCR signals.
We did not include mammals in the metallurgy analysis. We have some markers like C1 markers as the lab, so we can try on the sample. But for the 12S that we've been using, we also detect some other organisms like ungaral limbs in the sample. So.
All right, well, with that, we are actually heading to our breaks a little bit early. So thank you very much for joining the session. And let's give a round of applause for all speakers.
We're about to go into our keynote speaker now, Dr. Per Stenberg from UMIA University.
Thank you very much. So first of all, thank you very much for inviting me here. I'm really enjoying this conference. A lot of interesting talks, so I'm looking forward to the rest of it as well. Okay, so I'm not from around here, I'm from Sweden, and I'm going to talk about an eDNA project that happened by accident, that I just stumbled across. So I'll talk you through that. I work at Umeå University, which is located, maybe I can already show this one to us. So this is Sweden, if you don't know. I'm from a university in northern parts of Sweden, about the 64th latitude. And I work at, I work at two different places, Umeå University and then at the Swedish Defense Research Agency, half and half. What I talk about today is collaborative efforts between those two agencies. At Swedish Defense Research Agency we do, or at the unit where I work, we do biodefense stuff, related stuff. And at Umeå University we do ecology stuff, but on the same type of data. So, in Sweden, and also these systems exist also in other countries, but in Sweden we have currently 6 active aerosol monitoring stations. And they were set up, the first one was set up in the end of the 50s, and then the rest of the stations came up in early 60s. And the reason to set them up was to monitor radioactive fallout during the Cold War. So basically to be able to monitor nuclear test bombings that were going on. And they put up the first station in Stockholm in '57, and then just a couple of months later they discovered an increased signal in radioactivity, which they can then with aerosol models backtrack to an accident in a military facility in Windscale, I think. So then they realized that it was very sensitive, so they set up more stations. And for example, in '86, in fall '86, when the Chernobyl accident happened, we first saw increased levels of radioactivity in some Swedish nuclear power plants, and there was a question whether there was a local accident or so. And then they analyzed the data from these filter stations, and they could triangulate and accurately pinpoint it was exactly from Chernobyl. And it was only then when the Soviet Union admitted that they actually had a small accident. So it's a very sensitive system. And at each of these places there's like a small container-sized building. On the outside there is a silica-based filter, which is basically HEPA type filter. It goes down to 0.1 microns. But the cool thing is that the rest of the building on the inside is just a big fan pumping air or sucking air through the filter 24/7. And each week, so the filter is up for a week, and then during that week, more than 100,000 cubic meter of air passes through this filter, so 100 million liters of air. So each sample represents an enormous amount of air. And then every week, the filter is exchanged. They send the filters for analysis of radioisotopes, and then after measurement they compact the filters into small packs and store them in airtight containers. And this I heard about when I, in 2014, started to work part-time at the Swedish Defense Research Agency. And as a new employee, I got to tour the different facilities and see what we're doing. And then I heard this talk about these filter stations. And I was like, wow, this is really cool. But then when I heard it was silica-based filters, and I'm old enough to remember that the professors that taught me how to do DNA analysis as a PhD student, they were working before we had all of these tons of expensive freezers. So what they would do, they would extract their DNA, they would dry it into a silica filter, put it in an envelope in their office, and it would stay for decades without any cooling or anything. So I thought, maybe there are some other stuff in these filters that could be interesting to look at. And at the time my group was doing research in epigenetics, genomics, so we were used to genome sequencing, transcriptomics, chip sequencing, high-C sequencing and all of that. So basically throw massive amounts of shotgun sequencing on stuff to learn stuff. So I thought, why don't we throw massive amount of shotgun sequencing at these and see what happens? So we got a couple of filters to test, and we sequenced those. So now a lot of you work with air, so this is maybe not a big surprise for you, but what is in air? I would say that the simple answer is everything. If you sample enough air and you sequence it deeply enough, you see everything that has a reference genome, of course, because 50 to 70%, depending on how we do it, is completely unknown because of lack of reference genomes. But we see bacteria, we see fungi, we see plants, we see animals, we see birds, we see fish, we see moose, we see intestinal parasites in moose, and you name it. Everything that is reasonably abundant and has a reference genome, we can see. So that was in 2016 when we got those first results, and the natural reaction was like, is this really true? Can you get that much diversity in air? So we did a lot of confirmations, and since we do shotgun sequencing, we just started aligning the reads to the different reference genomes, and as you can see, these are some examples of different organisms, and we have a really nice, in most cases, even coverage of the reference genomes. Here there is a gap in the Drosophila because this is a mitochondrial genome that we map to, and that's the control region where you can't map reads uniquely. So, but otherwise, it seemed really to be true. So the next question was, okay, so we take some fresh filters and sequence it a lot, we can see a lot. But what about this insane archive? Because they have saved every single weekly filter from all these 6 stations back to the early 1960s. So can we really go backwards in time? Because that would be amazing. Because these eDNA technologies around today, they're great, but they're modern. So if you start a time series now, it's going to take you a few decades before you know proper ecosystem dynamics and how it responds to different environmental factors like climate change or land use change, whatever. So we checked if historical archive samples would work. This was just a very small scale metabarcoding based study over a few years from northern and southern Sweden, but basically it just showed that phenological patterns that we know about are preserved in the filters. So we compared with pollen data and some other things to just show that archived filters seem to preserve DNA very well. So then we made our first long-term study. So we started by sequencing filters from northern Sweden. So then we talk about subarctic region, basically the northern limit of the boreal forests in Sweden, because we thought that, well, the further up we get, maybe we see more evidence of climate change because temperature differences are larger up there. So we sequenced every week, more or less every week, I should say. We skipped the winter weeks when there's tons of snow up there, because then we don't see a lot of DNA in the air. We do see some tree-living fungi and bacteria, but most of the interesting diversity is not present during winter. And then we sequenced every second year, which turned out to be a really, really bad decision because it makes modeling much more complicated, but well, we were no experts when we started this. Anyways, so we had weekly data for basically 34 years. And as you know, with season, different organisms release more DNA, for example, plants when they flower and so on. So we early on realized we could look at phenological patterns, and we recently published one paper where we looked at moss phenology, because I presented this at a conference in Sweden and met with a guy that's, I guess, Sweden's leading expert on mosses, and he said that, well, the last thorough investigation of moss phenology was in 1850s, when there was a guy that spent two decades, German guy spent 2 decades out looking at individual mosses and look at when they sporulate, because in microscopy it's really hard to see the difference between different spores if you just collect them in air, so to say. So we thought that maybe we could use your data, and we did. So we showed that for a number of common mosses in Sweden, their sporulation period had moved between 4 and 6 weeks earlier in the season during this 34-year period. Another common question about eDNA is it actually quantifiable? Can you say anything about real changes in abundances using this data? And of course, as people have pointed out already, we will never measure absolute abundances. We'll never count the number of crows in an area or something like that. But we can see relative changes across time. And remember, since we're doing shotgun sequencing, we're sequencing the total diversity with a single method. So we have the total number of reads is some kind of a total biomass that we can relate to. So if we do that for a few birds that we had in our data, and these were the only birds which we could find external monitoring data from this region, because it's a quite remote region, so we didn't have a lot of external monitoring data to compare to. So we modeled their time series trends across the time series and compared it to external monitoring data, and I would say it's quite good. It has they compare approximately the same level as two different types of traditional monitoring methods would compare. So I think it works really well. But again, this is just shown for these birds, so we need a lot more work to show that it also works for other organisms. Then, with air, another big problem is where does the DNA come from? That's a big issue, because we have working together with the guys that do this backtracking of radioactive signals. We have really good models we can work with. And to the left there, in A, you just see the average catchment area across these 34 years. And the different colors is just a tenfold difference in probability for particles arriving from those areas. But we can do that for every week individually, that's not a problem. But the big problem with those models that are really accurate is that you need to input the particle properties into the model, like the size and the aerodynamic properties. Otherwise the models can't compute. So that means that for example, there in A, we have computed that for 22 micrometer particles, which is birch pollen. So it's probably very accurate for birch. But when it comes to, I don't know, moose, cod, what's the particle size of those? We have no idea. We have a lot of plans to try to figure it out, but we haven't done so yet. So to look at where those signals comes from, we need to use much more simpler models, the so-called trajectory analysis, where you look at the corridor where the wind has moved through during the period before when the filter was up. And then we can just overlay all of those, and we can basically weight them by the strength of the signal from that particular species. So if you do that, you can see that cod DNA in Kiruna mostly comes from the north, seems like the North Atlantic. If you look at moose, it seems like it mostly comes from northern Finland and more central parts of Sweden, which matches known moose population densities. There's quite little moose up here, although there is some. And we can also do that temporally. So if we look at, for example, reindeer during the warmer weeks, the signal is stronger up in the mountains, and that's when the Sami people, the indigenous people in Sweden, they have their reindeers up in the mountains where they normally live. But then during winter, they usually migrate the reindeers down to the coast for feeding when there's too much snow in the mountains. And then we can see that the signals switch direction, basically. This study we very recently published. We also did some analysis on the general ecosystem changes, and you can would be happy if you read the paper, but very short summary of the results is that we can see over this time period a very drastic decrease in biodiversity, no matter how we measure it. And most of the organisms that go down are actually linked to old growth forests or really old forests. So we did not see what we expected to find. We did not see a lot of impact of climate change, but there is another very big human impact which causes basically all of the patterns we see, which is forest management practices. So we can see changes correlating when they change how they manage forests. Right. Now, I skipped all the technical and maybe boring. For some it's boring, but maybe for some of you it would be less boring. I will just briefly mention, so we got the data in, I think we got it early 2019, and we published it now December 25. So it took us many years to go from data to publication, because it is highly complex data. We also spent about a year just figuring out how do we do DNA extractions, because we have a mix of particles in our filters. Some of them are pollens and spores, where DNA is highly protected. Some of them are like cell fragments or even naked DNA. So how do you extract DNA? If you treat it too hard, so you open spores and pollens, then you will destroy all the East-access DNA. If you treat it too mild, then you don't crack the spores and so on, so you don't see the plants when they flower and so on. So we spend a lot of time there, and basically what we're doing, we do a series of consecutive DNA extractions of the samples, and then we pool them in the end to get as high diversity as we can out from the samples. Then the sequencing matters a lot, turned out. We did a lot of pilot experiments, just how do we sequence, because we wanted to do shotgun to avoid PCR, but you still have a few rounds of PCR when you make your sequencing libraries. So we wanted to minimize that, so we tested what you're looking at there without going too much into details. This is just a 2-cycle difference in the library preparation. It's 7 and 9 cycles, I think. And then that difference in cycles doing the library completely shifts your composition of, in this case, we're looking at GC-rich versus GC-poor bacteria, the relative proportions of those in the data. So that completely shifts your data. So we have to do exactly the same number of cycles, exactly the same DNA extractions for all samples, which meant that we put a threshold on if it has less than 20 nanograms of DNA, we don't sequence it, because we want to have exactly the same input for everything, so we are as consistent as possible. But on average, since there's so much air, we take about 5% of the filters, and that gives us on average about 200 nanograms of DNA, so it's quite significant amounts of DNA. Then the database, we want to compare to everything, so we have always constructed our own databases. If you do shotgun, I would strongly recommend not using the MT or MR databases. They are highly problematic to use. A lot of bad stuff in there, both contaminated genomes and wrongly classified taxonomy and all of that. You can never avoid all of that, but if you put some effort in constructing your databases, it will be insanely much better. But they are big, so we couldn't fit them into normal computers. We had to find really high-performance computer resources to use. The first PhD student that worked on this, he came back when we got the data a couple of weeks later and said, I was trying to blast the reads, and from this time series, I don't remember exactly how many, but 200 or 300 billion reads that we got out. And he said, I was trying to blast them, and I calculated it would take 700 years. So I was like, okay, so we need to solve that. So we are not using alignment-based classification of the reads. It's just impossible with this amount of data. So we use Kamer type, Kraken and the source. And that is excellent because it's really fast, but it makes a lot of shortcuts, and it also has a lot of assumptions about you need to know the taxonomy and so on. So you get a lot of problems when using Kamer-based classifications. For example, you see directly that up in northern subarctic Sweden, you have a healthy, stable population of white rhinos and Tasmanian devils, which kind of stands out as problematic. So we spend lots and lots of effort how to clean that up, and I think we now have a very good approach. We're using machine learning algorithms that we train on a lot of metadata from the classifications, because you can see how different K-mares across your reads are distributed taxonomically, and there's a lot of information there that you don't get if you just look at the standard output, but you can have softwares like Kraken output a lot of information that is really useful. So we trained the machine learning on that and known observational data. which is another thing important to then realize that we do that only on basically larger eukaryotes, because that's where observational data is available. So these models will be biased against small stuff, microbes and small fungi and so on. So we don't know how well the models perform on those. Okay, so then we have classifications, and then, as you probably also know, all sequencing data is compositional. So if you have two species in a system that is completely stable first, and then one of them increase, then the other one will, per definition, decrease. So if you just take your raw data and you look at trends in it, you will see a lot of spurious correlation that has nothing to do with biology. So you can't just use your data as it is, because it exists not in Euclidean space, but in simplex space. So we need to do transformations of the data, log ratio transformations, and we need to apply proper mathematical models that can deal with this compositionality. So not just standard correlations. But I'd be happy to talk about more details on this if anyone is interested in a coffee break or so. Because we spent, I think, almost 40 person years on just getting from the data to results that are useful. So for the rest of the talk, which is not so much longer, I will just briefly talk about some other stuff we're doing with this data and some new data sets that we have. This is one fun observation we did during the analysis of this Northern Sweden data, is that we didn't see a lot of directional trends, some of course, but what was really standing out was that a lot of organisms seems to show oscillating trends over time. And one of the strongest such signals was wood cypertrophs. If we look at wood cypertrophs, they seem to have some 8-year cycles in abundances. And we are trying to figure out what is causing that. We haven't, I can't say we have proven that in any way, but it seems to be a very strong correlation between these cycles and these large-scale pressure systems in the North Atlantic. So there is a teleconnection pattern called East Atlantic, West Russia, and that seemed to have very similar 8-year cycles. But again, 8-year cycles, then 34 years of data is nothing. I mean, we need hundreds of years to actually statistically prove it. So it's a hypothesis that seems plausible, but we're not sure yet. And we don't think it's a direct effect on the fungi. We think it's an indirect effect that these pressure systems affect the winter conditions, which affect the host plants, the spruce and the pine that are dominating up there in Sweden, and then secondarily affects the wood cypertrophs. Another fun thing you can do if you sequence everything is not just look at taxonomy, but look at diversity. This is 2 spring weeks from a year when birch flowers, and you align the reads to the birch chloroplast. You see that for a couple of weeks of data we have, well, a couple of thousand X coverage of that genome. So we can look at SNPs or SNP diversity. Of course, we can't assemble and look at haplotypes or things like that, but we can look at frequencies of SNPs. And we have done that for a few trees, and we're now trying to publish a paper where we look at spruce and pine genetic diversity across time, and we can clearly see a decrease in pine genetic diversity. There are a bump there towards the end of the time series, which correlates well with when they changed the breeding programs and introduced new genetic variants, but it has since then also continued to decrease. And again, caused by forestry, of course, because northern Sweden is just forest, but it's monocultures of pine. It's very little natural forests. In one project, so since we now have all the data, the problem is we misreference genomes. So we have in different projects, for example, one we collaborate with lichen experts, and we're right now sequencing 100 new genomes of lichens, so we can go back to the data and track them through time. We are looking at a lot of different forest pathogens. Sadly, if you send in an application saying that I want to understand how ecosystems behave over time, I want to understand how they're impacted by human activities, they say that, nah, you don't get the money. But if I say that, well, we're going to sequence hundreds of samples for millions of Swedish kronor, hundreds of thousands of dollars to look at 5 or 4 fungal pathogens that are harmful for Swedish forestry, they say, yes, here's the money. And then we do that, and then we use 99.9% of the rest of the data for more fun stuff. But yep, you have to adapt to reality. At Defense Research Agency, we're using this data also to look at different pathogens. This is the Swedish favorite pathogen when it comes to human pathogen. It's Francisella, and it has the species Francisella tularensis that we have also here in Northern America. It's a traditional potential bioweapon, and both US and Soviet Union did a lot of bioweapon production based on this organism. Anyways, I don't need to go into details there, but one thing that we can do is we found one single spike in the data which was really strong for Francisella tularensis, the pathogen we're interested in. But most organisms, we have very low coverage. I mean, we're talking about far below 1x coverage of most organisms, so we can't really say exactly the genotype for most organisms if they're not very common. So to do that, we went back to our DNA archive, because we have all DNA samples saved, of course. So then we made an enrichment panel for Francisella. We pulled out the sequences from the DNA pool that were enriched in Francisella, and we sequenced those, and then we could easily reconstruct exactly the genotype that produced that spike. So that's just a proof of concept. We're also spending a lot of time right now trying to use our data to investigate this dark matter vDNA. So everyone knows we lack a lot of reference genomes, but where do we lack them the most? For many organisms, we know that these are species we know about, but we don't have a reference genome. That's fine. But for a lot of microbial diversity, and especially fungal diversity, we don't even know the species are there. So we're trying to use the data to estimate what are the taxonomic groups with mostly undetected diversity, so to say. And this is just a small-scale study we did on Francisella, but we're now trying to expand that on microbes. With this data, I always had a goal, but I think most people think of it as an illusion, but I still have hope that we can use these time series to not just map how things are changing, but also to understand how things are interacting. Because it would be really cool if we could use these time series to investigate how species interact in these ecological networks, and see if those interactions change with different pressures from the environment. And so far we've only done it theoretically. So we've just submitted a paper where we've done theoretical modeling of species changes with food webs. And then we're using these relatively new but really cool mathematical models called empirical dynamic modeling. So you're basically trying to see, can I predict future behavior, or do my prediction of the future behavior of X, is that improved by knowing how Y changes, but not the other way around, so you can find directional interactions. And on simulated data, it works really well, but it really remains to be seen if our data can be used for this. But I'm excited to see that. I guess, yeah. We did a small pilot some years back, also starting to look at the more southern parts of Sweden, because in the southern parts of Sweden is where all the food is made. That's where we have all the farms, because up in the north where I live, it's too cold. We have potatoes and some milk cows, but that's basically it. But in southern Sweden is where basically within that circle more than half of Swedish food production is happening. So we thought we should look at that area. And for this study we just looked at pathogens affecting agriculture in Sweden, but very long story short, if the pathogen has been observed by the Swedish Agricultural Board in the area and it has a reference genome, we do see it. And it seems like the damage levels roughly correspond to the EDA amounts we see. But that small scale, it was just 9 weeks during a single season, so we can't say much more than it seems to work. So now we're expanding that together with a lot of Swedish agencies involved in safe food production. We have a research center that we just started on threats to the Swedish food system. It's not just what we're doing on the biology side, it's a lot of other sciences involved, but I won't go into that. But what our contribution will be in this is that we've now sequenced 520 consecutive weeks from the last 10 years from this southern station. And for this project, the goals are to understand abundance and phenomenological trends, what are the drivers of those, and if possible, predict future trends. But of course, also we'll use the same data for other ecological questions. But within this framework, we'll focus on threats to the food system. And the ultimate goal is to be able to detect if we have an outbreak of something, is it natural or is it not natural, to basically understand if we're under some kind of an attack. But just we have just started to look at this data, it's very new, but this is what the raw data looks like for some rust fungi that's problems for grass type of crops. We can look at the animals or crops themselves, of course, and different microorganisms associated with those. But the good thing with this data now is that we're in southern Sweden, populated area of Sweden, where there's actually people, not like where I live, we're very rural areas. So here we have lots and lots of data to compare to, which is going to be great, so we can validate our data much more. So for example, for crops, we have GPS coordinates by the meter, exactly where each crop has been for every year throughout the time series. So we have now been able to use that to improve, for example, our source estimates much more accurately. So we have used trajectories again with similar approaches I talked about, but now we're adding on a lot of other information. We're adding on what are the land types that the air has moved through. We can calculate decay functions because it's not, of course, the same probability that the particle coming from a trajectory very far away has the same probability of actually reaching the filter as if it was originating close by. So that has really improved our ability to pinpoint where the signal comes from. So now we can, and we're just starting to do this, but basically now we can, for all the thousands of species we have in the data, we can get we can calculate with our models a long-term trend, we can calculate what is the seasonality, has that changed, we can calculate how different organisms are associated with different land types and so on, and where the most hotspot source areas are. And this just happens to be one species that is problematic in Sweden, an invasive species. And we had this is just starting, but very, very, very general. You can see that over this time period, what is doing great is fungi. It's getting warmer and wetter in Sweden, so fungi is doing great. A lot of other things not. If we zoom in on insects, we can see that most insects are not doing so well in the last 5 years or so. But I don't think that we'll have to wait for another meeting. But just lastly, I still think that this type of archive is really useful. And of course, there are other archives of EDNA. You have the sediment cores, we have ice cores and all that, and that is great. You can go back even further in time. But the type of taxonomic and temporal resolution you can get in this type of data, I think it's really fascinating. And again, this system is not unique for Sweden. So you have them here. They are part of this nuclear radioactive fallout monitoring system that exists everywhere. The question is just, have you saved them? Because I think the Swedish agencies are not excellent in many things, but one thing we're excellent in is saving stuff. We never throw stuff away. We have insane basements where we just... And I mean, these filters, no one understood why they were saving them, because they measured radioactivity already, so why save them? And they tried to force them to throw them away many times, but they were like, no, it might come into handy sometime. So you check, maybe you have local stations here you can use, and maybe the filters are there. I encourage you to do that. But we have a long time going before we have done everything in Sweden. We just sequenced these time series that you see there. So we just have 18,500 filters to go, and that just needs another $50 million or so. So if anyone has that laying around, I would be happy to. So just end by 2 weeks ago, we were out playing with our new drone in the southern Sweden area to document the surroundings. And with that, I thank you.
Thank you. Amazing talk. I have so many questions that are about bioinformatics that I will grab you at a break because it will take forever. But one of my main questions is how many reads are you targeting per sample to kind of get at that robust community for the shotgun?
So of course it varies a bit, but somewhere between 150 and 600 million reads per sample. Higher than 150 base pair reads.
Also, really love this talk. Has any of your data actually led to conservation action or any kind of mitigation efforts based on what you found?
No, not yet. We have to also remember that what we are doing is really regional scale surveillance. I mean, we will never, even if we improve the models further, we will never be able to say that these couple of hectares have changed like this. Then you have to have much more small-scale, local type of sampling to be able to say anything like that. What we have seen and what is a bit of a problem for us, going back to the first talk this morning about ethics, we have had some challenges when we publish in what do we disclose, because as you know, there are false positives in this kind of analysis, and we can sort them out, but there's a lot of manual work for each individual species to align them to reference signals and really see that it's an accurate signal. So we can't do that for everything. So what do we then publish? Because a lot of the things that potentially is there is controlled pathogens that there are. You have to report them to the agencies, and they have to do different things to mitigate these things. And so it is a bit of a challenge in what you choose to publish. So we try to only publish what we can really, really validate, and then of course we publish the raw data, but then we don't publish sort of all intermediate data to scare people unnecessarily, because I'm validated. Yeah.
Hello, thank you very much for your talk. I was wondering, how do you evaluate, or what variable do you use to monitor abundance trends? You said that you were...
Well, so I mean, of course you have the relative amount of DNA changing across time, but before that gets into a time series, I mean, the models need to take into account wind directions, what kind of land types the air passed over, what's the temperature, humidity, wind speeds, because you get more aerosols when you have strong winds, and there's a lot of things you need to build into these models, and also we have to take into account the seasonality, because the seasonality is always the biggest variation in the data. So you have to do a lot of modeling before you get to actual abundance change across years. But it seems to work well if you do it properly.
I meant to use the number of reads, or what?
Well, that's the raw data, but then there is a lot of mathematical steps between that and the final results. And it's a lot of computations in between.
Second big question is, how do you get rid of false positives? I mean, just by geography, or?
No, so we use this machine learning approach where we feed the machine, the model with hundreds of variables that we can extract from the actual classification of the reads. So I talk, for example, about how are all the camers that are identified in a specific reef, where are they classified in the tree of life, and how far, what are the taxonomic distances between those, and there's a lot of things you can feed into this. And then we train it then on known species that are observed in the area. So we take a 100-kilometer circle, and we say that if it has been observed at least 10 times in there, it's true positive. And then we say that if it's only been observed, but it has been observed many times outside of, I don't know, like maybe it was 10,000 kilometers, I don't remember exactly, but far away. So we make sure that it's possible to observe, but it has never been observed close by. Then we say it's a false positive. And then we train the models with this metadata on that. And then we have test data. So we normally achieve about 95% accuracy and 75-ish recall. And I think that's really good. Of course, we have 5% false positives in there, but they're dominated by closely related species to the species that are actually there. But this local Swedish species doesn't have a reference even, but let's say a North American version has. So it's a false positive, but it's almost.
All right, Bob, I'll leave you a question on lunch so we can ask her then. Please join me and thank you for the
Our next guest speaker is Brendan Daisley. Good morning, everyone. Really great to be joining you here today. So I'm a postdoc here at the University of Guelph, and my work really focuses on bee microbiomes and understanding them. And through this work, we've really learned that bee microbiomes can tell us a lot about the surrounding environment as well. So this is also the first eDNA conference I've ever been to. So really excited to kind of see the different methods everyone's using, and it's really exciting to kind of, see the different science being done in the same way. And I think we share a lot of the same methods, and hopefully some of those things will be appreciated and transferable. Okay, so when we talk about biodiversity loss, we typically think about visible organisms, right? But for example, the vast majority of plants and animal species in various different ecosystems. But underlying that macroscopic reality is microbial foundation. We're now realizing that a lot of microbial biodiversity is at risk and basically declining. And this is, in most of the cases, we don't even know what microbes we're losing because we're not tracking them, right? So this is really important because microbes play crucial roles in things like ecosystem function as well as host development, innate immune functions, and overall adaptation on an evolutionary timescale. So this is especially crucial when we think about insects, where microbial symbionts can support rapid physiological adaptation within changing environments. So one group, where this becomes particularly interesting is bees, because they sit at the interface of so many interactions within the environment, and also they have a really unique coevolution with both plants and microbes. So, for example, bees forage across complex landscapes and interact with a range of mediums, including flowers, water, soil, and air, as well as many other insects within the pollinator assemblage. And because of this, they integrate environmental signals over space and time. And in essence, bees essentially represent nature's original environmental filters. And this is why bees have often been discussed as biosensors or bioindicators in the context of environmental contamination. Their bodies and hive products can reflect exposure to things like pesticides, heavy metals, and other pollutants. So most of this work has really focused on chemical bioaccumulation. However, bees do not only encounter environmental pollutants, they also interact with healthy and diseased plant communities, healthy and diseased pollinators, floral microbes, fungal spores, bacterial communities, viruses, and environmental DNA. And notably, recent work has shown that flowers can act as shared microbial hubs where basically pollinators can deposit, acquire, and exchange microbes. And this includes not only good microbes, but also many microbes that can cause disease in insects, as well as many plant diseases for which pollinators can act as vectors of. Thus, this means that bee-associated microbial signals may tell us more than just about the individual bee. They have potential to reflect host health, the plant communities present in a given area, and also the local pathogen burden, as well as broader environmental exposures. So this raises ultimately an interpretive challenge, though, is basically how do we tell what microbes are true host-associated symbionts and which ones are environmental signals. So from an evolutionary perspective, we know that bees have co-evolved with a select set of so-called core gut microbes, which play a really crucial role in supporting their health. And specifically, corbiculate bees are those bees with anatomical structures referred to as pollen baskets that help them collect pollen, share a core set of microbes known as the corbiculate core. So that includes 5 species there, and also more that aren't listed. But beyond that, over time, they've well, one thing also to mention is that this is believed to be shared at the point of origin of eusociality. And since that time, basically different lineages of bees have acquired other bacteria that are specific to those lineages. And we can use these differences to essentially separate bees based on their microbiome composition. So you can see in principal component space here, looking at the microbiome of these different pollinator species, honeybees, for example, green, bumblebees in blue, and stingless bees in red, show very nice clustering, and they're based on these specific organisms that have co-evolved with the bee. So we can use that to kind of parse out, microbial signals based on their host. So what's not shown here is that bees are also consistently exposed to plant and environmental associated microbes as well. So the microbiome we ultimately observe reflects a balance between conserved host-associated symbionts and also environmental microbial inputs. So to move beyond individual case studies, we conducted a global meta-analysis of bumblebee microbiomes using publicly available sequencing data. And this work came from a really excellent PhD student I advised, Christine McPherson. She gave a talk on some of this yesterday. But essentially the goal here was to ask whether broad environmental and anthropogenic stressors leave detectable signatures within wild pollinator microbiomes. So to do so, we analyzed publicly available 16S microbiota datasets from bumblebees around the world. And across 814 bumblebee samples from 15 studies, we examined stressors including pesticide exposure, captivity, land use, and other environmental pressures. And overall, we found really clear shifts in microbiota composition, and these were often marked by reduced microbial diversity or a complete loss of microbes. So what you're seeing here is just essentially complete absence of these microbial genera in these studies, depending on their various exposures of interest. So certain anthropogenic exposures of pesticides, glyphosate, and various heavy metals lead to a complete loss of these environmental signals. And also, when you take the bees out of those natural environments, when they're in captivity, they also lose those environmental signals very clearly. And it just kind of adds to the theory that many of these agrochemicals that are being kind of released into the environment are having a negative effect on microbial diversity, which can ultimately pick up in the bee microbiome here. So importantly, we show that these effects were also context dependent, depending on host species, geography, exposure type, and baseline microbial community. Obviously, in some cases, anthropogenic exposure didn't always lead to a loss of those microbes, and in a lot of those cases, it's because the bees were also, partly in natural areas, so they kind of had that ability to rewild themselves, if you will, because they were in this shared environment. So ultimately, the main message is that anthropogenic stressors can reduce microbial biodiversity in bumblebees, but the outcome depends heavily on ecological context. So relevant to these findings is the missing microbe hypothesis, which posits that a sudden loss of microbial diversity in the gut of animal species in particular can lead to significant health consequences. So this has been most well studied in human populations, and specifically in relation to transgenerational antibiotic exposure. So since around the 1950s, our grandparents, our parents, and now us, we've all been exposed to antibiotics at some point, right? And this has led to a loss of gut microbes at a population level. We all are missing microbes. And basically the thought is that that loss of microbial diversity linked to antibiotic exposure has led to many chronic metabolic diseases that are surging in industrialized populations, including hypertension, type 2 diabetes, and a variety of cancers. So applying the missing microbe hypothesis to bees, we found that systematic depletion of key symbionts can drive distinct states of microbiota disruption or dysbiosis. And specifically, we found that not only antibiotics, but also many agrochemicals such as herbicides, fungicides, and also not shown here, insecticides can disrupt their microbiota. And importantly, bees appear especially vulnerable to these disturbances because of their own reductive evolution. So essentially, their genomes have shrunk, which has led to depauperate immune and detoxification pathways. And specifically, gut colonizing lactic acid bacteria are known to be major modulators of bee health and play crucial roles in supporting immune function, pathogen defense, and the metabolism of environmental toxins. So one example connecting microbiome science with macroscopic conservation efforts come from our collaborative work with Wildlife Preservation Canada. And in short, the bumblebee, or WPC's Bumblebee Recovery Program focuses on conservation of several at-risk bumblebee species. They essentially collect individuals from a range of sites across Ontario, breed them in captivity, and then release the reproductive females or gynes back into nature. However, conservation efforts have been failing, in particular for Bombus turricula. And this is due to a variety of microbial imbalances and also disease pressures. So to investigate this, we performed 16S microbiota sequencing on some of their bumblebee samples and found that Bombus turricula, samples shown in green here, all cluster away from the other bumblebee species for which conservation efforts were working. So it showed us that there is, a kind of a direct link or a microbial signal associated with these bumblebee species that essentially were having difficulty with conservation efforts. So looking deeper into this, we also found that the Bombus turricula had a reduced overall microbial diversity, and this was characterized in particular by a reduction in lactic acid producing bacteria shown here. And essentially, those are those beneficial symbionts that bees rely on for immune function and detoxification, right? So without them, they can lead to disease susceptibility and a variety of other health consequences. So our working model is that these bumblebees may have lost these beneficial symbionts through their differential behavioral patterns or anthropogenic exposures. So again, going forward, we're essentially performing collaborative studies to test whether recolonizing the bee turricula with the missing lactobacillus species found in corbiculate counterparts can improve health and ultimately conservation efforts to save the species. So in other words, if microbial disruption is why a population fails, then microbial restoration may need to become a part of the conservation plan towards population recovery. And these examples, lead me to my broader research question we'll be discussing today, which is, can bee-associated microbial and eDNA signals be used to understand host-health, environmental exposures, and biodiversity change? So this question really has two parts, right? And the one is, can bee-associated microbes tell us something about the health of the bee itself, right? And then the second question is, can those same samples be used to tell us about the environment bees are moving through? And that includes things like pathogens, floral diversity, plant-associated microbes, and broader biodiversity change. So to study this at scale, honeybees have become a really useful high-throughput model for us. And so this is because honeybees are widely managed. They can be repeatedly sampled, and they're also connected across agricultural as well as natural habitats. Their colonies essentially provide a practical sampling unit for large-scale surveillance. And in peak, so demonstrating this in peak summer, a single bee is estimated to visit around 50 to 100 flowers per day. So you look at a colony with 60,000 foragers, and this equates to approximately 3.6 million flower visits per day from a single colony. So it really, demonstrates the breadth of the sampling capacity bees are capable of. And this is up to a 10-kilometer foraging distance from the hive as well. So essentially, using honeybees, we can explore multiple layers of biological signal, including bee-associated microbes, entomal pathogens, plant-associated fungi, pollen and flowering plant diversity, and also baseline microbial diversity across terrestrial habitats. So essentially, this makes honeybees in particular a really good model for us to ask the questions we're interested in. So today I'll be focusing on three main research areas. The first is how honeybees can be used as pathogen surveillance tools. And so related to this topic, we performed a field trial in California, which represents a central hub where millions of colonies are shipped each year to pollinate almond crops. In short, the reason why so many bees are moved here is due to the fact that modern cropping methods don't adequately support enough habitat to sustain pollinator populations year-round. So essentially, the honeybee colonies are moved around the country in kind of a musical chairs fashion according to the bloom periods of different crops. So obviously, there's a lot of issues with the way we could do current agriculture, and it's beyond the scope of this presentation. But one particular issue is that this approach acts to concentrate pathogens from all across the country into a kind of a centralized zone in a really intensive, short period of time. So accordingly, around 1.5 honeybee colonies are imported to California each year, and from that, approximately 80,000 of those die, primarily due to disease pressures. So this makes, in essence, this makes California a hotspot for entomopathogenic disease transmission, and also, which I'll be discussing, phytopathogenic disease transmission. So in our observational field study, we tracked 33 colonies across 2 major pollination sites and collected samples at monthly intervals across a 24-week period. And instead of using qPCR to look at specific pathogens of interest, we took an untargeted approach by performing 16S and ITS sequencing to look at essentially all of the bacterial and fungal species there. And overall, we detected a total of 848 unique bacterial ASVs and over 3,000 unique fungal ASVs, demonstrating the sheer diversity of captured through bee foraging in this pathogen-dense area. So reflecting this, here's the compositional bark plots of the most abundant bacteria and fungal communities present. And so what you're essentially, I know there's a lot going on here, but essentially what you're looking at is each bar for each of the graphs represent a single colony, and then within each bar, the colors represent the unique composition of what bacteria or fungi that are there. So overall, you can see that the bacterial compositions were more stable between the hives and at each time point and also over time. However, they show a slight seasonal trend towards increased lactobacillus and bifidobacterium species over time. So those again are in light blue and in darker blue, and those are those beneficial symbionts that honeybees and other bee species rely on. Conversely, for fungal communities, we see greater hive-to-hive variation at each of the time points, and also greater overall diversity and lower stability over time. And you can see by the final time point that there's actually a complete change in the overall structure of the communities. So another way of looking at these differences is by visualizing them on a heat map and normalizing the abundances to the start of the study. So again, there's a lot going on here, but the main things is that this is the bacterial group now shown at the species level, and the fungal group now shown at the species level as well. So if you look at time point 0 for both, it's normalized at that time point, and then over time, species can either increase, shown in red, or decrease, shown in blue. So you can see, really clear kind of chunks of, groups of bacteria, for example, that have increased, including the lactobacillus and bifidobacterium species that I mentioned on the last slide. And these are those, good microbes. So it suggests the microbiota is kind of showing an improvement over time, potentially recovering or stabilizing after disruption from transport and or agrochemical exposure. And another thing that, what's really neat about this is we detected a number of different entomopathogens and phytopathogens. So they're shown by different colored stars here. The red ones are the insect pathogens, and the green ones are the plant pathogens. So if we had used a targeted qPCR approach, we wouldn't have been able to even detect these because we didn't know what we were looking for. And a number of these plant pathogens I'll be discussing in a little bit, play a really important role in not only agriculture but also environmental health. So, stemming from these, another point too is basically we saw linkages between how the microbiota changed and the abundance of important bee pathogens, including Listococcus plutonius, which decreased over time with this improved composition of the microbiota, as well as a fungal pathogen, Ascosphaerus apis, which also decreased over time. So, it sort of suggested to us that there was a linkage between microbiome composition and disease load. So we wondered, from an experimental point, could we modulate the microbiota or could we target it to experimentally reduce these pathogens in a controlled way? So we did that in another set of experiments. And to test this theory, we essentially performed the same 24-week field trial setup, but this time we supplemented colonies with a probiotic LX3 solution containing 3 different strains of lactic acid bacteria. And we have done a number of other studies on this to show that they specifically modulate honeybee immunity and also detoxification pathways. But essentially, for simplicity, they're good bacteria, we supplement the colony, and we did so in 2 different ways. We tested 2 delivery mechanisms, either through a feeding process, through a pollen patty, or through just simply spraying an equal number of colony-forming units on the hive surfaces. And then we also include vehicle controls for each of those groups as well. So we first looked at Meliscoccus plutonius, which is the bacterial pathogen that causes European fallow brood in honeybees, as well as a number of different native bumblebee species. And we found that this probiotic supplementation significantly reduced the abundance of this pathogen in both of the delivery formats. And interestingly, this spray-based application actually showed stronger efficacy in delivery than through the pollen patty supplement. So when we then looked at Ascophera apis, which is the fungal pathogen associated with chalk brood disease in bees, we showed that in this case, feeding the probiotic in the pollen supplement had actually no effect. And yet the spray reduced pathogen abundance by over 90%. So follow-up experiments, we did a number of follow-up experiments I'm not going to show, but they essentially showed that this stronger effect was likely due to direct contact between lactic acid bacteria and the fungal pathogen. So mechanistically, this appeared to be pH mediated, with the beneficial bacteria essentially helping to acidify the local hive environment, which restricted the fungal growth. So we thought, if these probiotics could reduce these important bee fungal pathogens, what could they do to the plant pathogens that were also present in the hive? And so we looked at that, and this is showing essentially the overall cumulative decrease over the 24-week period induced by the supplementation of the LX3 or the increase in some cases. But for the most part, the supplementation actually ended up decreasing most of the fungi present. So our findings demonstrated that LX3 could significantly reduce the burden of several plant-associated pathogens, including Boitres cinerea, which is the causal agent of gray mold, and also it reduced Alternaria alternata, which is a widespread fungal pathogen associated with leaf spot and other diseases across 380 plant species, as well as Fusarium longifundum, which is the causal agent of canker disease in almond trees. So in the later case, we saw an absolute remarkable reduction. It was over 30 log-fold change reduction. And this could partly be explained by the fact that bees were foraging in an area with a dense number of almond trees. And essentially the level of this pathogen was really high to start. So nonetheless, the LX3 spray-based method led to a near complete eradication of the pathogen of interest over the 24-week field study. And so this suggests that probiotic supplementation in bees might be a way to reduce transmission of not only bee diseases, but also certain plant diseases relevant to agriculture and environmental sustainability. So more broadly, these findings connect to the flying doctors concept, where bees can be used as beneficial microbes or biocontrol agents as they move through flower crops. So in our case, we found that bees were not only useful as samplers, but also samplers of pathogen burden, but also potentially as delivery vehicles for microbial biocontrol. So after showing bee-associated samples could be, capture broad range of pathogen signals, we became interested in a related question: if bees, collecting microbial signals from plants they're foraging on, could they also be collecting the plant DNA themselves through the pollen? And this led us to ask whether honeybees could essentially be used to assess flowering plant diversity. And to test this, we designed a 2-year observational study across multiple crops and regions with focal crop systems including apple, corn, canola, blueberry, and soybean. And colonies were essentially categorized as either exposed or non-exposed on whether they were within the 1.2 kilometer threshold of the focal crop of interest. The logic was fairly straightforward here. If bees are good samplers of nearby plants, then we should be able to detect stronger crop-associated plant eDNA signatures in the colonies located near those crops. So to measure this, we used a metagenomic approach here rather than a targeted amplicon approach. So once again, we collected bees from the trapped colonies. We dissected their guts, extracted DNA, and then performed shotgun sequencing instead of ITS and 16S sequencing. And the goal was essentially to allow untargeted detection of plant DNA in the samples. And since bees collect pollen, in particular, from plants, and pollen contains approximately 20 times more mitochondrial DNA than somatic cells, we expected this to be particularly useful for taxonomic classification purposes. So what we found was that plant DNA was definitely informative, and we captured a lot of diversity, but not all in the ways we initially expected. So overall, we detected 274 plant species across 120 colonies. So what you're seeing here again is each bar is representing a single colony, and then within the bar showing you the composition of different plant species that were detected. And along the top here is a key showing what region the colonies were in, the crop type that they were most close to, and whether or not they were considered exposed to that crop type. So, contrary to our initial expectations, samples clustered really more strongly by region than focal crop type. So we saw essentially minimal separation between exposed and non-exposed colonies. So, take the BC samples here in light blue, foraging near high bush blueberry. We see minimal clustering between the kind of the red and green groups, which would indicate whether they're exposed or not exposed within that 1.2 kilometer threshold. And we don't see really much clustering, nor do we see much clustering in canola crops in Alberta. And so this trend was kind of consistent for the most part. There's some obviously outliers there, but it suggested to us that the bees were essentially not simply sampling the dominant forage nearest to the hive. Instead, the plant DNA signal appeared to reflect essentially broader regional foraging patterns and plant community composition. And one important limitation I also wanted to mention here is that plant identification relies heavily on reference databases, and several presenters have already mentioned this. But plant species are basically the plant species that are absent or poorly represented in databases such as NCBI are obviously going to be difficult to detect with when there's no references. So we suspect that if their databases were more comprehensive and we could actually map all of the reads to plant material, then we might be able to see a better separation between these two groups. But for now, those databases don't exist, so this is what we got. Nonetheless, we ultimately saw the same patterns when we looked at the data in ordination space. When we visualized the samples via principal component analysis, we found no difference in year-to-year variation, nor did we see any difference in the distance between focal crops of interest, so whether they were exposed or not. Instead, the results essentially suggest the bees may be actively selecting forage floral resources rather than passively sampling what is most locally abundant. This led to one of the more interesting ecological interpretations from the study, specifically that geographical region was the strongest predictor of overall sample clustering. But at the same time, we found that bees are essentially not passive samplers of plant diversity. Instead, we interpret this to mean that they basically provide a behaviorally filtered interaction-weighted signal based on the fact that they sought out other plant species beyond the dominant crop, including native and non-crop resources at each of the different sites. So while this could be considered a limitation in a lot of ways, if your goal is to essentially catalog every plant in a given region, it could also be considered a strength and highly informative if the goal is to understand which plants actually are being used by pollinators. So this obviously has relevance for biodiversity monitoring, habitat restoration, and conservation planning when thinking about pollinator systems. So far, I've discussed how bees can be used for pathogen surveillance and also profiling of flowering plant diversity. And the next area I'll be discussing is basically whether bees can be used as indicators of overall terrestrial biodiversity. And the goal here, and I've alluded to this a little bit through my talk, but the goal here was to essentially develop a baseline. Right now, when we look at microbial diversity in bees in different areas, we have no baseline. So it's hard to tell if they've lost microbes, right? So we're really concerned with a lot of the agrochemicals that are being used because a lot of them induce antibiotic-like effects, right? And they're not being regulated in any fashion, and no one's looking at it. It's, we could be experiencing essentially a microbial apocalypse right now, but we have no evidence of that, right? So the idea, or to address this, we developed the Canadian Bee Gut Project, which is essentially a national scale effort to develop a baseline for microbial biodiversity across Canada using honeybees as biosensors. And the idea being that, basically sampled colonies nationwide, we can use that information to begin to build a mapping system of what microbial biodiversity looks like and also for future use and conservation planning. In other words, the Canadian Bee Gut Project is not just about describing the gut microbiome of bees, it's about using bees as a scalable platform for microbial biodiversity surveillance. So to achieve this, we used a citizen science project because we would have not been able to collect all these bees ourselves. And basically we teamed up with beekeepers across Canada, and after a major outreach campaign and recruitment effort, we sent volunteers a standardized sampling kit containing instructions, questionnaires, sampling tubes, and QR tracking tags to enable longitudinal monitoring of their hives. So beekeepers essentially collected the bees and then sent them back to us at various time points before and after winter each year. And then downstream, we processed these samples using DNA sequencing and to identify which microbes were present, and also an NMR-based metabolome approach to identify relevant chemical signatures. Ultimately, these data will allow us to build, that national scale atlas of pollinator-associated microbial biodiversity, and also link microbial and chemical profiles to geography, season, land use, and also, colony health as well. So far, we've collected samples from over 700 hives across 100 unique locations nationwide. And here's just some behind the scene photos of us preparing the sampling tags and sampling kits before sending them out to beekeepers, and also testing the QR codes with local beekeepers as well. So here you can see ultimately the national distribution of sampling locations across Canada, for which the Canadian Bee Gut Project collected. And notably, we're still processing the majority of the samples. So what I'm about to show you is just essentially some of the really exciting preliminary 16S microbiota data for this first several hundred batches of samples. So what you're seeing here is a heat map of the bacterial ASB prevalence across sampling regions. So each row represents a bacterial ASB, and then the columns represent essentially the sampling regions and the presence or absence of that ASB in that region. So even from this really early data set, we can already identify a really clear set of core microbes, right? Some that are present in nearly all samples, and then also some that are present in many of the samples, but not all. And so, coming back to the idea of missing microbes, these patterns are really interesting to us because, some of the inconsistently detected taxa may represent beneficial symbionts that have been lost or depleted from different lineages of bees across Canada. And at the same time, when we look beyond the core microbiota at the environmentally associated fraction of the community, we see substantial variation across regions, including some really fascinating location-specific microbial signatures in different provinces that might be related to various habitat ecosystem functions there. But one of the most important findings is that a large we also detected over 4,000 bacterial ASBs in total. So, highlighting the enormous microbial diversity captured through the project. But one of the most important findings is that a large fraction of these sequences didn't match to any known organism in public databases. In fact, more than 1/3 of the 16S diversity is expected to represent, novel species that have never been cultured or classified before. So this, although projects like this can generate vast insight, into microbial biodiversity, they also expose a major bottleneck, being that much of the microbial diversity we detect is still effectively unknown. So, this brings us to the concept of microbial dark matter. I think it was called eDNA dark matter in the last presentation, but nonetheless, the vast proportion of microbial life that we can detect through sequencing, yet we can't yet confidently classify, culture, or functionally interpret. So the next challenge then becomes, if sequencing reveals all of this hidden biodiversity, how do we actually make sense of it? So one of the ways we've been doing that is through developing niche-specific databases and developing other sorts of bioinformatic tools. For the purposes of today's talk, focused on bees, I'm going to be talking about the development of the BExact database that we've used to improve classification of microbial dark matter. So diving into things, let's consider the bioinformatic steps when analyzing a typical 16S sequencing data set. You can imagine, you've done your DNA extractions, you perform sequencing, demultiplex your FASTQ files, and you denoise your reads with Data2 or whatever other pipelines you're using. And now you have essentially a list of amplicon sequencing variants or ASVs from your data set. However, when you then go to align these ASVs to a universal database for taxonomic annotation, many of the ASVs come back as unclassified. And depending on the classifier tool you're using, you might also get back the lowest common ancestor, for example, the closest genus or family rank. But nonetheless, the main issue is that downstream, all of these NAs or unknowns are being grouped together. And this is despite the fact that they may be very distantly related from a phylogenetic standpoint. So this means you essentially no longer have phylogenetic consistency, and thus you're no longer dealing with monophyletic groups, and ultimately this can end up skewing your data set resolution. Oops, skip forward too much there. To address this issue, we essentially developed a curated niche-specific database and assigned designated placeholder names to those uncultured species representatives. And in brief, this process involved 3 main steps. The first was development, or the construction step, and this is where we essentially use keyword searches to compile all bee-associated 16S genes that are present in all databases that exist, NCBI, EMBL, and others, as well as authoritative references from literature sources. So at this step, we removed the low-quality sequences, dereplicated the reads based on prefix matching, and a number of other quality filtering steps. But we didn't cluster the reads to ensure that basically, they were still high resolution and compatible with ASVs. And the next step, which was the curation step, we essentially annotated sequences up to their lowest common ancestor using established sequence similarity thresholds and bacterial type strain information as stable points of reference. We then assigned placeholder names to the sequences remaining unannotated, and this was guided by maximum likelihood based phylogenetic inference to basically minimize branching conflicts. So you can imagine, you take 2 novel species that are within a novel genus. None of them have names. We basically assigned placeholder names to each of those species, and then if they were predicted to be within the same genus, we also gave them a shared predicted genus name. And this was done to ensure essentially, phylogenetic consistency and a rooted structure in phylogeny within these placeholder names. And then finally, we validated the database by performing in silico tests on over 234 million reads across 50 million bee species, 50 bee species. This was done on 32 real data sets and consisted of 36 eusocial and 14 solitary bee species. And then we ended up identifying essentially 4 novel order, 9 novel families, 81 novel genera, and 618 novel species. So, in practice, how does this actually help things? We compared the BExact database to several other databases that are commonly used and showed that BExact essentially strongly outperformed them all, including the leading database, Silva, that is used. So in summary, BExact has essentially reduced the amount of microbial dark matter by providing de novo taxonomy for 618 species. And more broadly, it's helped standardize how researchers track, classify, and communicate the associated microbial diversity across studies. And reflecting this, since its release, it's been cited by 55 studies and seen really good uptake by the community. Another key point is essentially that this approach isn't limited to bees, right? The same strategy can be applied to really any niche habitat of interest. So funny, if you're working on a niche habitat of interest with poor classification, I'd love to speak after this talk. And then that brings me to my overall summary for the talk, which is basically that bees represent useful biological samplers, and they integrate microbial, plant, and environmental signals across ecosystems. So, from a pathogen surveillance perspective, bee-associated samples can reveal diseases affecting bees, but also plants and also broader pollinator communities. They can be used for characterizing plant diversity in a given region. Pollen and plant eDNA can show which floral resources bees are actually using in different habitats, and this is behaviorally filtered. And then from a terrestrial microbiome's perspective, we're at the Canadian Giga project, we're showing that bee-associated microbes can provide insight into environmental microbial biodiversity at large, and also ecosystem change over time. So the next challenge, I think we can all agree with all the work we're doing, is interpretation. Linking sequence data to ecological context to understand which signals matter the most for things like health, conservation, and environmental monitoring. And I didn't discuss it in this presentation, but I think a lot of that is going to come down to developing better databases. In the last talk, put good emphasis on how useful in-house databases can be. I think we coming together, maybe we can start making better public databases through using existing sequencing data to essentially compile metagenome assembled genomes for things that we don't actually have the physical organism to get the genome for. But beyond the scope of this talk, I think that's kind of the future direction for a lot of our work and work in general. And with that, I'd like to thank all of the team that helped this work be completed, as well as all the funding agencies that we couldn't go without. And with that, I'd like to thank everyone and take any questions. I think someone beat you to it.
You call it the bee-associated database. I think it's really interesting. These are APIS, right?
Yeah.
And they're not native to Canada. I'm curious about the influence of the fact that we're using a non-native single species. How representative is it going to be of the 10,000 native pollinators, do we think?
Yeah, for sure. I mean, the reason we chose to use Apis mellifera is just simply because of the fact that they're managed, and there's so many of them per colony, and they're used, religiously all around the world. So we can kind of use them as a sampler. Whether or not those bees should belong in that environment is a whole other discussion that I fully agree with where you're going with this.
I was actually kind of going with the fact of I like curated in-house databases, but that one may be very specific to a non-native species in the wrong environment. So I call it the APIS exact database as opposed to the bee.
Oh no, the bee exact database had over 50 different bee species, including 36.
From the colonies, okay.
No, 36 eusocial and the rest solitary species. So it's a broad range of essentially all corbiculate bees.
Okay. Then the question I had about the database then was your dark matter tag set. I'm really curious. Did you ever find one which should be a core that we've somehow?
Yeah, for sure. I mean, even in the heat map from the Canadian Giga project, the core section I showed, if you look there on the right, a number of them are shown in red. They represent microbial dark matter.
And how do you know they're real sequences as opposed to some mutated version of a similar sequence? That's always my problem. Is how do you know it's new and not just error?
Yeah, like chimera maybe. Yeah, certainly. I think that's always a problem. I think shotgun sequencing help with that, because when you can generate complete metagenome assembled genomes, metagenomic assembled genomes, MEGs, you have a lot more certainty that that's a real organism rather than just a single barcode. Do you disagree with that?
No, I think there's a danger of over-diagnosing new things from noise.
Yeah.
I don't know how you draw that line, but.
Well, what I was saying is basically when you can make a MEG, a metagenome assembled genome from that dark matter, I think it's a lot more believable if you can form an entire circular chromosome. I think there's a lot of data to support that it is real at that point.
Yeah, have 2 questions. So just to clarify, everything that you're doing is based on bee gut contents, right?
Yes.
Okay.
So that's basically consumed environment,
Yeah. Okay. And when you talk about like decreasing the plant pathogen load that you see in the, like you're talking about like bee doctors, right?
Yeah.
This is assuming bee-mediated transmission of pathogens. It's not really, I mean, unless you do something like bee vectoring, you're not really decreasing the incidence of the pathogen on the plants that affect the plants, right? You're decreasing the bee transmission of that pathogen because the bee is visiting different plants, right?
Yeah, so it's a little bit different than the flying doctor's concept, but like it's in there. But yeah, the idea is that like we're lowering the amount of the plant pathogens in the hive, so it's like, and we found that actually, beyond just the bee gut, a number of other studies, like experiments we did, we showed that it actually reduced it from the hive surfaces too. So it kind of cleared the hive of those plant pathogens. And another, from that work, another kind of question that came from is like, are these plant pathogens growing a little bit in the hive? Like, are the hives acting as a vector, right? And so, the kind of cool thing that came from that was that the good bacteria we kind of sprayed the hives down with reduce the growth of those plant fungal pathogens.
So quick second question is, this bee Apis mellifera is a generalist as a species.
Yes.
But, so we think that we can, and that's a problem that I have as I work with Apis mellifera. But each one of the hives turns to become a specialist in what resources they go and harvest.
Yeah, like individually, you mean? Like individual hives?
Yeah, like as a species, we think about Apis mellifera as a generalist, and it is, right?
Yeah.
But when we look at the individual hives, even hives that may be close together, they kind of become generalists. And there's some studies that show that you can predict phenology of plants from harvesting, like for example with pollen pellets. But it's very random. Like the problem that I'm having is I want them to be generalists to kind of like survey through the whole season and see if we can actually, for example, detect invasive plants. But it tends to be very random and more associated with like phenological cycles of the.
Yeah, and I mean that's we've observed the same thing, and I think the way to kind of address that is just by sampling more colonies at a given site to increase. It's like kind of like one of the talks earlier mentioned, increasing the number of filters you put in a bat cave. It's like use more colonies at a given site, improve the likelihood that you're going to get a random sample of that. Within that close proximity sort of thing.
Well, with that, I think we need to thank our keynotes.